Good afternoon and good evening, everyone. My name is Stephanie Goslin, and I'm the Programs Director here with Ovarian Cancer Canada, and I'm so excited for our evening ahead. I would like to begin by acknowledging the Indigenous peoples of all the lands that we are on today. While we are gathering virtually, let's take a moment to appreciate the importance of the lands we each call home, and to reaffirm our commitment and responsibility in improving relationships between nations, as well as our own understanding of Indigenous peoples and their cultures. We're excited to be bringing together over 280 people this evening from coast to coast. Before we begin, I just wanted to go through a few housekeeping items. So we will be recording our event tonight, and that will be available on our website at the end of next week. And we also would encourage you to take advantage of our researchers and experts this evening by asking questions. You'll see the Q&A button at the bottom of your screen. If you can type your questions in there, we will get to those questions at the end of each session. Now, keeping in mind that we know you might have questions about your own individual experiences, unfortunately, our speakers won't be able to provide individual advice at this event, but will answer more generic questions. If you have questions that are specific to you, we would encourage you to talk to your healthcare team about that. So thank you, everybody, and we are ready to begin. Ovarian Cancer Canada is driving research further, faster, and leading an international movement to galvanize Canada's ovarian cancer research community. Just 25 years ago, there were only 3 scientists dedicated to the study of ovarian cancer. Now, because of Ovarian Cancer Canada's efforts to attract leading minds to this field and to provide the raw materials researchers need, Canada's ovarian cancer research community is 250 plus strong and growing. These scientists are among the most collaborative and engaged in the world, and we're happy to have some of them with us here tonight. We are so grateful to all of you for joining us for the Research Forum. Thank you for your support and joining our community. Events like tonight are made possible because of your support. If at any time tonight you would like to learn more about Ovarian Cancer Canada, our research, or how to support this work, you can visit our website at www.ovariancanada.org. So now I would love to introduce you to one of those amazing scientists, Ovarian Cancer Canada's very own Dr. Alicia Tone. Alicia holds a PhD in ovarian cancer biology from the University of Toronto, postdoctoral studies in ovarian cancer genomics at BC Cancer Agency, and more than 7 years as a scientific associate in the Division of Gynecologic Oncology at the Princess Margaret Cancer Centre. We are so grateful to have her as part of our team. Now over to you, Alicia.
Okay. Good evening, and thank you so much for being here. It is my honor to present an overview and brief update on OVCAN on behalf of the many scientists, clinicians, patients, and advocates involved in this national research initiative. This session will cover 3 things: how OVCAN came to be, our research priorities and bench-to-bedside approach, and some highlights of progress in our first 3 years of this 5-year initiative. In 2015, in consultation with communities of patients, clinicians, and researchers, Ovarian Cancer Canada created a 3-pronged research strategy named OVCAN. This strategy had a clear single aim over 5 years: to improve the outcomes of women diagnosed with ovarian cancer through funding for research into better treatments. 4 years of dedicated and collective efforts by Ovarian Cancer Canada, women living with the disease, family members, and other advocates across the country followed. In 2019, the Government of Canada took bold action and entrusted Ovarian Cancer Canada with an unprecedented $10 million investment into ovarian cancer research to improve outcomes. In early 2020, OVCAN was launched. So we have since taken the federal government's $10 million investment and grown it to almost $14 million by advocating with provincial governments in Saskatchewan and Nova Scotia and working with partner research institutions such as Iroquois and the Cancer Research Society. Funds are allocated to projects within 3 research priorities: support of key national resources such as tissue banks, patient engagement activities, and personnel required for project management. An amazing 90% of funds are allocated to research projects. So the first research priority focuses on development and characterization of experimental models that closely match the disease in patients and represent the diversity of histologic types and molecular subtypes of ovarian cancer. Many of these models are derived from tissue generously donated by patients, and they enable us to learn about how each type of ovarian cancer starts, how it progresses, and how it can be stopped. This important work provides the tools needed for scientists to discover and test new potential treatments in a living system prior to testing in patients. The second priority focuses on preclinical testing of potential new treatments in these highly representative research models. Patients have played a key role in selecting the most promising treatments to be funded within this priority area. The third priority focuses on personalized clinical trials taking place across Canada that focus on testing of promising new treatments in patients diagnosed with different types of ovarian cancer. This is important as response to standard chemotherapy varies based on factors such as ovarian cancer type and molecular alterations, such as specific gene mutations in the tumor or individual. Identifying patients who are not likely to respond to standard treatment and offering them effective alternative treatments remains one of the most important goals in the clinical management of ovarian cancer. These three priorities spanning from bench to bedside all feed into our ultimate goal, which is to improve the outcomes of women with ovarian cancer. So now on to some of our progress. So within priority 1, our goal was to build a gold standard set of ovarian cancer research models that could be made available to the entire Canadian ovarian cancer research community. To achieve this, we created an OVCAN Models Consortium, a pan-Canadian group of scientists with relevant expertise. We have funded 23 projects to date using a uniquely collaborative decision-making process, whereby open discussions on collective funding priorities are held through working group meetings focused on different types of models. Importantly, funded projects on research models will set the stage for future discoveries, whether diagnosis, prevention, or treatment for all types of ovarian cancer. I'm not going to go over the details on this slide, but I wanted to show the names and faces of the many Canadian researchers we are funding to complete this important work. For instance, on this slide, these individuals from Quebec and Ontario are all creating models specific for high-grade serous cancer, the most common type. We also have funded researchers from BC, Quebec, and Ontario to support projects dedicated to less common types of ovarian cancer, including but not limited to low-grade serous, clear cell, endometrioid, and rare types such as small cell carcinoma of the ovary, hypercalcemic type. These researchers from Quebec and Nova Scotia are being funded to develop tools that will facilitate discoveries regardless of ovarian cancer type. So within priority 2, our goal was to identify and prioritize the development of up to 8 novel treatments to be tested in preclinical studies. We've in fact exceeded this goal, funding 13 studies in collaboration with funding partners, IRACOR and Cancer Research Society. And importantly, this includes 5 projects dedicated to less common types of ovarian cancer and funding of an early career investigator to invest in the future of ovarian cancer research. This is showing you the faces of all of the priority 2 funding recipients from 4 provinces, including BC, Saskatchewan, Ontario, and Quebec. All of these projects hold great promise, and we can't wait to highlight results and next steps as they progress. Finally, within priority 3, our goal was to fund 4 to 5 early phase clinical trials using a personalized or precision medicine approach. We wanted to ensure that any studies we supported would be national in scope or at least have multiple sites across Canada, build on Canadian science, include studies on samples collected throughout the trial to learn as much as possible, have meaningful patient engagement, and have a large potential for impact. So we are very happy to share that we've again exceeded our goal, funding 6 clinical trials through open competition with international expert panels and patient reviewers. And up to 331 Canadians with ovarian cancer are expected to be enrolled in these studies combined. So I'm just going to show you a few things about these studies that we have funded. So the NEO trial, led by Drs. Amit Oza and Stephanie LaRue in Toronto, is testing the impact of using a PARP inhibitor prior to surgery in women with platinum-sensitive recurrent high-grade serous ovarian cancer. This trial includes 5 sites in Canada from Ontario, Quebec, and Alberta, and 44 patients have been recruited with analysis of tumor samples underway. The ES2 trial, led by Dr. Diane Provencher and Helen McKay from Toronto, will test the impact of using a PARP inhibitor in combination with another drug that activates controlled death or apoptosis pathways in tumor cells. This study is gearing up to open very soon and will recruit 30 women with high-grade serous cancer from 3 sites in Canada. The CX5461 trial, led by Dr. Amit Oza in Toronto, is testing a new small molecular inhibitor, clearly called CX5461, in patients who become resistant to PARP inhibitors. This trial is now open at Princess Margaret in Toronto and the CHUM in Montreal, and is aiming to recruit up to 30 women with ovarian cancer of all types. And finally, the REVOLVE trial, led by Dr. Stephanie LaRue in Toronto, is truly personalized medicine in action. This trial is set to open very soon and will use genomics in tumor samples to determine the best next treatment for patients who become resistant to PARP inhibitors. Up to 100 patients with recurrent high-grade serous or endometrial cancer from 9 sites across Canada will be enrolled. So the 2 most recently funded trials are being led by Drs. Anna Tinker and Yvette Drew from BC Cancer. Preparation for these trials are underway, and you will hear from both of these women in the coming sessions tonight. But briefly, Dr. Tinker's study focuses on the identification of predictive biomarkers in women with low-grade serous ovarian cancer, one of the less common types. Dr. Drew's study will test the impact of combining a PARP inhibitor and 2 other drugs in newly diagnosed stage 4 patients with high-grade serous cancer. Projects led by 3 Saskatchewan researchers, Dr. Laura Hopkins, Shaina Lee, and Miriam Elheki, are also underway. Projects include creation of a biobank, tumor testing, and ovarian cancer drug prediction program, studying perspectives of patients and clinicians in Saskatchewan, and the effect of iron supplementation on survival. We are also funding an integrated program in Nova Scotia, with 3 projects being led by Dr. Stephanie Scott, Jeanette Boudreau, and Robin Urquhart, now underway. These projects will combine to result in expanded ovarian cancer biobank, a detailed study of immunology and genetics in patients throughout treatment, and a better understanding of ovarian cancer care in Nova Scotia. So very importantly, women living with ovarian cancer are integrated throughout all aspects of OVCAN and our work in general, keeping the voice of those with lived experience at the forefront. Engaging ovarian cancer patients and their loved ones as partners in research reflects our philosophy that scientific research can be enhanced by valuing and uplifting the unique perspectives of those affected by the disease. So the role of our patient partners in research program is to complement and maximize the impact of research being done by Ovarian Cancer Canada supported scientists. Our team consists of 20+ women from 7 provinces, all but one of whom have been personally diagnosed with ovarian cancer. The team is led by co-led by Donna Pepin, highlighted here, who also serves as patient advisor on the OVCAN Governing Council. The full patient partner team attends monthly meetings where they hear from scientific experts, participate in ongoing education, and prepare for engagement opportunities. These are just some of the faces of this dynamic team. So examples of activity that our patient partners have been involved in include participating in international and national review panels, in small interactive sessions with researchers, as panelists at National Ovarian Cancer Canada Symposia, integration as full team members on OVCAN-funded clinical trials, consultation on funding proposals, surveys, and educational tools. You can see there's a lot. Collaborative research grants, participation in collaborative research grants with us and external partners, and invitations to speak about the successes, challenges, and best practices for patient engagement at national and international conferences. So I want to wrap up by saying that this program is truly a national effort with engagement from the clinical, scientific, and patient communities from 8 Canadian provinces. Names in bold on the left are those experts serving on our governing council, which acts to ensure that all money spent aligns with our overall goal and mission of OVCAN. So with that, I'll thank our funders and partners and pass the torch to the next session.
Great. Thank you so much, Alicia. It's amazing and so exciting to hear about all the great work happening. So thank you for that brief overview and introduction. The next session is one that we get a lot of questions about, Clinical Trials 101. Dawn Richards is the Director of Patient and Public Engagement for Clinical Trials Ontario. Dawn has a PhD in analytical chemistry, and it was her own diagnosis with rheumatoid arthritis 15 years ago that started her journey to combine her passion for science with making the most of her diagnosis. Tonight, Dawn will be interviewing Trish Osborne from Palgrave, Ontario, and Joanne Lindsay from Toronto, Ontario. Both are Teal Sisters living with ovarian cancer who have firsthand experience as clinical trial participants. As well, we'll be joined by Dr. Alison Ross, Director of Disease Information and Education here at Ovarian Cancer Canada. So Dawn, I'm going to turn it over to you.
Great. Thanks a lot, Stephanie. And it's really my pleasure to be here tonight with all of you. I'm also really pleased to hear where the OVCAN initiative has come. I had the pleasure of helping out in the early days of OVCAN with some of the patient engagement activities. So I'm so thrilled to see the success that's been achieved with that today. So just a brief introduction to add to what Stephanie graciously provided already. As she mentioned, I'm the Director of Patient and Public Engagement at Clinical Trials Ontario, or CTO. And what CTO is an organization that's primarily funded by the Government of Ontario, basically to help Ontario become a better place to do clinical trials. And we do that 3 ways. One is to help streamline the processes, to help people do clinical trials, basically set them up faster and really get them up going so that people can participate in them, but they're still ethical and of high quality. We help promote Ontario as a place to do clinical trials. And then I do work like this, which is helping engage patients and the public with clinical trials, either on clinical trial teams, like you just heard about with Alicia, or as sessions like this, where I sit down with people who have participated in clinical trials, and we try and bring knowledge to a greater audience so that you can make the best decision for yourself around potentially participating in a trial or simply learning more about what the options are. So that's a bit about me and the organization that I'm here on behalf of today. And I'm really honored to be here with Trish Osborne and Joanne Lindsay, again, who have participated in clinical trials, and they've graciously allowed for me to interview them, and they'll be sharing some of their experiences, and hopefully I can add some context onto their experiences as well. We'd also invite you to either type questions in the chat or the Q&A. We can see if we can answer those as we go along, and if not, we'll make sure that we've got a bit of time at the end of the session before we wrap up to try and answer your questions as best as we can. So I'm going to ask both Joanne and Trish to say a few words about themselves and also share what you're comfortable with about the clinical trials that you've each participated in. So maybe I'll pass it over to you first, Joanne, and then Trish, I'll ask you to add on next to that. So go ahead, Joanne.
Thanks so much, Dawn. It's so nice to be here with you. My name is Joanne, and I was diagnosed with ovarian cancer in February of last year, 2021. And I have low-grade serous carcinoma. I started in a clinical trial after surgery in April last year. I was in a clinical trial that was looking at the standard of care for low grade, which was chemo, 6 rounds of chemo followed by letrozole or letrozole itself. And I was randomized to the stream to do chemo first. And so I did 6 rounds of chemotherapy, and unfortunately, it really affected me. And I had a tough time with very many side effects of chemo and needed a lot of support in that. So at the end of that, my doctor, who was the principal investigator, the PI in this clinical trial out of Princess Margaret Hospital, deemed me unfit to continue because I was just, I had many issues going on. So I was dropped from the clinical trial, and then I did start on the next stage, letrozole, 4 or 5 months later. So I will always wonder if that delay has caused any problems for me, but that is something that we will never know about. I was, I'd be happy to go into a clinical trial again if there was one that I would be eligible for. Let me pass it over to Trish.
Thanks a lot, Joanne.
Thanks, Joanne. Thanks, Dawn. I was diagnosed with high-grade ovarian cancer in March of 2019. I went on to doing chemo. I had the tumor taken out and everything, and I started my chemo right away. The month May I started my chemo, and I ended up starting right away on the trial too. So my trial was Bev and Durva. Extremely long names, so I just used the short form of them.
That's fine. Yeah.
And I started that. I did the 1 round of chemo, and then they started me in on the trial right away. Now the trial took me a lot longer than I expected. I finished my chemo, the 6 rounds, and then I didn't finish my trial until September of 2021. So it is a long time, but I've always said it's so well worth it to me. There was some side effects, probably not as much as maybe you, Joanne. I do have blood pressure and I have thyroid problems now, but I've always said I'm 6 feet above, not 6 feet under. So I look at that as a good thing. So if that's the little bit that I have, I'll take it. I find myself tired a lot in my bones. I find it's probably the job I do now. So my bones, I found, have changed a lot. And I don't know if that's chemo, if that's the trial. And yeah, I can't say enough about the trial, and I can't say enough about Princess Margaret. So.
Okay, thanks a lot to you both for sharing that. Joanne, we've already got a question in the Q&A, and I think it kind of makes sense if you're up for answering it. It was, so you left the clinical trial because of your physician's concerns about it, and you still would consider being in another clinical trial if you were eligible. Are you able to share your thought process around that for the group?
I think clinical trials are really important in healthcare research. In fact, I know they are. I'm certainly happy to participate because I know that we've gotten to where we're at with treatments because of other people volunteering to be part of clinical trials. So I guess in some way I consider it to be part of my social responsibility. And the other part of it, and one of the reasons why I was happy to enroll in the clinical trial in the beginning, was that it would allow me to be monitored more closely. So I know that that meant there would be more appointments and more tests, and I'm not sure what else, but it made me think that I would get better care because I was monitored more closely. And that was very valuable to me. So it was worth having extra appointments. And fortunately for me, I live not very far from Princess Margaret, so it was not onerous at all to go to extra appointments and have extra, for example, have extra CT scans compared to what the regular standard of care would be. And for me, that was a real bonus of being part of a clinical trial. I felt well looked after because of it. I feel well looked after even without it, but I'm not monitored quite so much, which, as Trish says, it's better than some of the alternatives. So you learn to live with that and work with it.
Okay, thanks, Joanne. And Trish, I saw you nodding your head a lot when Joanne was talking about, feeling like she was monitored more closely and those sorts of things. Would you like to add to that as well?
Yeah, I feel very much of that. Like, I actually have a CT scan coming up at the end of this month. It's every 3 months. It's an hour and a half a day off work, and then I go on the Thursday, and then that Monday I go see the clinical trial, the team that I kind of call as my second family, under Dr. LaRue, and my nurse and that, and they're amazing. Any problems you have, you email, they're on to you. That they don't even email you back, they'll phone you back. So I definitely say that with Joanne too, that it was well monitored. During that whole time of even the 2 years while I was on it, I had a special phone. And I had to actually, update everything. It was a survey. So I had a survey that I had to do and constantly hand that in when I would come in and write everything down. Everything was recorded. Everything was like to the T. Even if I had a headache, it had to be recorded. If I felt a little bone pain, it had to be. Ended up getting some sores on my gums and that. It was a trip into Princess Margaret. To me, it's well worth it. Like, it's so well worth it, the trial. And Joanne said she would live closer. I didn't, but thank God with my husband and that he would take the time off, and you worked as a team, and that's what gets you through.
Okay. So another interesting question has come up that I think, we would probably explore as well, but maybe I can add a little bit of context. This person has asked if you were worried about being part of the placebo or control group, and that possibly having a negative impact on your health by not getting treatment. So I just would like to chime in that Joanne, it sounds like you were probably in a phase 3 study because there was a standard of care and you were in a randomized study, which is usually a phase 3 type study. I think something that I would just like to bring attention to with the audience is that when there is a standard of care for a disease area, it's not ethical for people to be offered placebo. So the comparator would be standard of care versus the new therapy. And that's something that a research ethics board would look at when they would review the protocol for a clinical trial. If that is a concern for people, that's definitely something that you should ask the study team about. So I just wanted to bring that to people's attention. But did either of you have concerns about that or ask about that when you were discussing the potential to be in a study with your oncologist?
I did talk with my oncologist about that, and I, I was reassured that it was, the standard of care was chemo for 6 rounds followed by letrozole. And the trial was looking at whether the chemo could be dropped and people should just go directly onto letrozole. So I, when I was randomized to have chemo, in some way, I was a little bit relieved,
Okay.
which, when in retrospect, I think is ridiculous, but because that was the standard of care, but it caused me a great deal of problems. I might have been better off on without chemo. The kind of cancer I had, low grade serous, it doesn't. It's known to not respond well to chemotherapy.
Okay.
But at the same time, I think I might have felt like I was not doing something that maybe I should have if I had not gone on chemotherapy. So, it's hard to know. I mean, we can't go back in time and test that hypothesis about whether I'd be better off. But anyway, I'm on letrozole now, and I'm tolerating it fairly well. So I'm not discontent in any way.
Okay. Trish, did you want to add anything? No.
I wasn't too worried if it was a placebo or not. I think a lot of this has a lot to do with your mindset too, going into it.
Okay.
When I was told that I had cancer, I just assumed they're going to take it out and away I go. I had a trip booked so a month later, but no, it was Dr. Bouchard, who was my surgeon, said, really think you should try this trial.
Mm-hmm.
So I walked over and I signed the paperwork. The paper is thick, and the nurse is like, are you sure you don't want to take this home and read it? And I'm like, nope. My doctor says it's good. It's good. Like, I guess I just put the faith in Princess Margaret just because what they stand for and the trial. And I know they're not just going to give it to you. You have to want it, which I did, but you also have to have certain criteria. I do know. And obviously I did.
Mm-hmm.
So, yeah.
So you met the criteria for a study, and your
Yep.
oncologist thought you were probably open to that discussion. Yep. You had the discussion and you made your decision based on a lot of faith in them and their knowledge. Joanne, yep. What was it like for you? I think you also said that your oncologist approached you about participation.
I was trying to remember, Dawn, but
okay.
I think that even before I had surgery, my oncologist said that I might be qualified or eligible, or I think eligible is the term she used for a clinical criteria. Trial. Mm-hmm. Yeah, and then so as I was leaving the hospital, one of the doctors said, "Oh, we want you to take this drug when you leave, and take it every day." And then she came back and said, "Oh no, we don't, because you might go in this clinical trial." So what she was offering me at that time was the letrozole. Now, if I'd started that, it would have made me ineligible for the clinical trial. So I waited until they had done the the medical and biological workup to see what stage I was at and all this. And then I spoke again with my oncologist, with the research coordinator, They both spent quite a bit of time with me and whoever was with me in my appointment. They did give me a whole bunch of papers to read.
Yeah.
And I did take them home and I did read through them. But I do other work in healthcare, and I have a great deal of faith in research ethics boards and clinical trials. And I know that all of that has happened in the background. I didn't sign immediately, but I really had no hesitations at all to get involved. The clinical trial did kind of find me because my doctor, my oncologist, was the principal investigator. So that's probably not the experience that everybody has. But I had no hesitation in signing up for it. And I was offered all possible opportunities to learn more about it, to ask questions about everything. I remember at one point even asking my doctor, would she recommend this to somebody in her family? And she said, if you were my sister, I would recommend you do this. So that was kind of the final I don't know, like that made it clear that yes, I would do this for sure. So
Okay, that's really helpful. And I guess what you've both been talking about in quite a bit of detail, partially is the process of informed consent. And you both went about it in very different ways to make such a personal decision. One piece is signing that big form, right? Which is, all the things that are expected of you, potential benefits and risks that are known, who you contact if you have issues, all those types of things. And then you further delineated on this, Joanne, and you have too, Trish, that you ask questions along the way. So informed consent isn't just you sign a document, off you go in the study, but it is about asking questions along the way and making sure that you're still comfortable with being in the study, you're still comfortable with what's expected, the site visits, all of those things. And it is important for people to also know that, if at any time you feel like you need to stop and withdraw from the study, that is possible. I mean, that happened to you, Joanne, on the recommendation of your oncologist. And sometimes people have to undergo tests or, specific, it might be a scan or it might be a blood test, et cetera. And that's generally to make sure it's safe for them to leave the study in the state that they're in at the time. So I just wanted to bring that to people's attention. And then you both also talked about the study kind of found you because your oncologist approached you about it and realized that you met the so-called criteria to be in the study. And in oncology, that is often the case, that people are approached to participate in studies. In some other areas, people, they might be treated by a family physician who's just not as knowledgeable about the space, et cetera. So they might, look at a database like clinicaltrials.gov to find a study, or they might approach an organization like Ovarian Cancer Canada. So there are different ways to get into studies. You both had a very specific experience, but I just wanted to let the audience know about that. And another thing that people are asking, I'm noticing in the chat, is knowing whether you should be in a clinical trial or not. And I think that, again, is part of a personal decision if you feel that's something you'd like to explore, or if maybe your physician has come to you about that. But there's not really any specific time that is the right time sort of thing. But we have touched on here that depending on the study, you do have to meet specific criteria to be in it. Like you said, Joanne, your oncologist kept you off of a specific medication so that you would be eligible for the study, et cetera. So there's lots of little nuances about things that are important for people to know about. So another thing I just wanted to go back to, and you know, you both didn't have major concerns about participating. And sometimes people do get concerned about the safety aspect of studies. So I don't know if there's anything that you haven't already shared that you wanted to share with people about who might be concerned about safety, in addition to asking lots of questions about things. Any comments that either of you have about the safety aspect at all that you'd like to share?
I wasn't concerned about safety. I was on the clinical standard. I was randomized into the clinical standard. So
Mhm.
I think if I had not been if I had not gone on chemo and gone right on the letrozole, I think I might have always thought, I wonder if I should have been on the chemo.
Mm,
So you know, that didn't happen. So safety was never a concern for me because I was on the gold standard at that time.
okay.
But I
Yeah.
think Tricia was a little different.
Well, no, I never felt unsafe or anything. There were some like just side effects, but that's side effects on anything. And like I said, the team I had and the, you know, the whole team there were very, very, very professional, very caring. Um, and 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 you know, just considerate. Like any time I was in, it was, you know, I like to go in the first thing in the morning because I could get in there before the traffic and then get out of the city before the traffic.
Mhm.
And they accommodate it, you know. Um, you know, they had the parking and we submitted our parking and there was, you know, fun that helped us, you know, we didn't have to pay for, you paid for your parking up front, but then you got reimbursed on that. But I I've always said I'm 6 feet above.
Right.
Thanks to them.
One thing, Trish, you also mentioned about was that you were surprised that the clinical trial took as long as it did. And so I wonder if you wanted to comment a little bit more about that, and if you continue to have follow-up visits at all. And then I'm gonna pass it over to Joanne to just comment as well. If you would have remained in the study, if you could share a bit about how much longer you would have been in the study.
Yeah, thanks. I was supposed to finish in June of May or June of 2021. And when I ran, no, it was, yeah, May of, May of something like that. And I was supposed to finish then, but then they asked if I would stay a bit longer to do, they thought it would only be like 36 treatments, but it ended up being like 42. And of course you miss some certain ones too because of your blood work and everything. So it was longer than I expected. But I was okay once, you know, my husband and I talked, and I was a little angry at first because they didn't prepare me enough, I guess.
Hmm.
But once I talked to my nurse and we got things, you know, she explained everything to me, then I was like, yep, no problem. And I stayed on it. It's to help, unfortunately, future patients. You know, I wouldn't want to see me go through the whole trial and now I can help another woman. You know, that's my biggest thing on this trial is now they're able to take whatever I have and you know, run with that and learn from that. So
Okay. Joanne?
And I think, I'm sorry, Dawn, I really can't remember, but
That's totally okay.
the letrozole, I think I understood at that, you know, when I was signing up for the clinical trial, that this is a drug I would take every day for the rest of my life. So for some reason, it pops into my head that it was like 3 years that I happened to be the first Canadian registered in the trial. So it was really just the very beginning of the trial. And for some reason, I think it was going to go on for the monitoring was going to go on potentially for up to 3 years, but I might be wrong on that. So the difference for me is that I now the standard of care is to have CT scans every 6 months instead of every 3 months. But it turns out I had a CT scan that showed them something of concern. So now I'm actually on a 3-month schedule. So,
Okay.
and I think that will fluctuate depending on what they find.
Right.
So again, I don't feel that I've, you know, I do feel that I have excellent care. I don't feel anything is missing from the care that I am getting.
Yep. So it's been interesting too, talking with both of you. You talk about, for you, it's been important to participate, not just because this might help yourself potentially, but you've also talked about helping other women. You've talked about the fact that you know, it helps to contribute to the evidence that, you know, ultimately helps potentially change healthcare or, you know, changes how decisions are made. Is there anything else that you wanted to add up to that about why you feel that trials are important, or why or how you feel they help either you or other people?
Well, I do know that trials are important. They're looking at the clinical care guidelines, and those are guidelines that you know, that all doctors are supposed to use. Sometimes you know, they don't, but you know, it's something that that medical practitioners of all sorts should pay attention to. And so, you know, that's important. I mean, to continue to understand, I know that low-grade serous carcinoma is not, it's not so common as other types of ovarian cancer. And for me, I think that means that there's less research done about it.
Right.
So this is a project that looks specifically at low-grade cancer, and I felt like I didn't feel like I had an obligation. I felt like I wanted to contribute to it, to the research that would maybe help me, but it would definitely help other people along that were diagnosed after me. So I guess it's like, I just sort of, you know, I see it as a part of my own sense of social responsibility, that it's a good thing to do. And it's good for our community of Teal Sisters, for example.
Awesome. Thanks, Joanne. Trish, did you want to add to that?
Yeah, I didn't realize how much, how many ovarian cancer stages they are. I was high grade, carsa, I can never say the long word. It was the two what you had, Joanne, but I was high grade. So
serous carcinoma?
Yeah, that's it.
Okay.
I had no idea that there was different stages. I didn't realize, you know, I had no knowledge of it. But to me, it was probably one of the best things that happened to me, because it has changed me so much, and it has made me aware of, you know, women, and to advocate for that now, to advocate for research, and to advocate for trials, and to advocate for, you know, to understand, women need to understand the different symptoms and everything of ovarian cancer, because I was like stage 3, if not stage 4. So yeah, it's
okay. Okay, and another thing, I'm watching the chat, there's a lot of commentary about myths about clinical trials. So some people see clinical trials as like the last ditch effort. And I'm certainly not hearing that from either of you. And so what would you like to say to combat that? And also, what other myths would you like to bust about clinical trials, given that, Joanne, I know that you work in clinical trials in another area as a professional. And so maybe you can draw on some of that as well.
Yes, so I do work in HIV research, but not specifically in clinical trials.
Okay.
It's slightly, but I'm very aware of all the clinical trials that do go on in HIV research and how important they have been in establishing new and very innovative medications for maintenance, that people, people take drugs every day for the rest of their lives, but the side effects are minimized by the newer treatments. And so clinical trials are ongoing in HIV. There is no cure for HIV, then many researchers are looking into that part of it as well. But clinical trials have developed and continue to develop very innovative treatment regimes for that health condition. So when I thought about a clinical trial in ovarian cancer, I immediately think this is, we're working towards better options for people in treatment. And, it's again, not a cure, but it's it's treatments that will have potentially fewer side effects. And I know in the trial that I was in, I had enormous side effects from chemotherapy, but I haven't had any from letrozole. So if that becomes the new gold standard, I could understand. Then I would have been better off in that, because I'm still feeling effects, some of the side effects of neuropathy, for example. So I'm a strong advocate for clinical trials, and I believe I will talk to my oncologist about options for joining another clinical trial in the future. I don't know if that will be a possibility because I'm not, I'm not really familiar with all the different kind of research in ovarian cancer, but I'll certainly be talking with my healthcare team about that.
Okay. Trish, did you want to add anything about, you said you weren't really very familiar with clinical trials in the first place. So did you have preconceived notions or thoughts about them beforehand? Or even, when you've talked to people about participating, have they said anything about that?
No, I wasn't worried about it. I did meet one woman that I would call it chemo daycare. And she was offered the clinical trial, but she turned it down because of driving. And I advocated to her saying, it's to me, it's so well worth it. It's so well worth it. I agree with Joanne. Like if it wasn't for us, 100 years from now, we may only be taking a few pills, for maintenance and stuff like that. The chemo kicks the living crap out of you, but I found with my pills, I didn't have that many side effects.
Okay. So another thing that you've both talked about is the fact that, Joanne, you live fairly close to the site where you participated. So that was like pretty convenient for you. Trish, you said you didn't, but you formed a team with your hubby and you guys decided this is what you were going to do. Is there anything that's important to share with people about that, about, some of the aspects of participation, or did you have to pay for anything out of pocket, or did the trial cover anything? Those types of things that might be helpful for people to know about?
I think I could have, I ended up taking a taxi often, but it was like often less than like $10 or less. So I didn't end up putting any claims in, but I think I could have. And then sometimes I would walk home from my appointments. So that would have, that if you live far away and you have to pay parking rates, I'm sure the parking rates in the hospitals downtown Toronto are pretty high. So I can certainly understand that. I don't know if they also paid mileage for travel, but I understand that once you're in a clinical trial, they pay for everything. So if there are drugs that you have to take, you get it for free. So for me, I'm over 65. So I basically get the letrozole for like, $4 for each prescription that I pick up. So so I don't know how much it would cost regularly.
Otherwise, you didn't have to out yourself, by the way, Joanne, but I appreciate that you did.
Like I said before, that we paid for our parking up front, but then you submit it and we got a check back afterwards. And
Okay.
it was like I would pack up, snacks and lunches, and because it was a whole day affair. When I was doing chemo and the trial, it was 8 hours a day.
Yeah.
You're there a long time. So you make sure that you pack for that. And I mean, chemo daycare, they have microwaves and, water and stuff like that. And no, I never looked at it as
Like a burden or anything?
No.
Okay.
No.
Yep. And so that's just something important people should think about. There's a comment in the chat about what's provided and, what you might have to give up, or your time or whatever is important for people to ask about. The other thing too that we sometimes get questions about is understanding the credibility of studies. And, a red flag would be being asked to pay for the study treatment, or being asked to pay like a participation fee or an entry fee type of thing. So those are things you should ask questions about if you are asked about that, but you should have a good sense of, the time commitment, what it may cost. And I know living in the GTA, if I drive downtown at midnight, it's going to take me 20 minutes. And if I drive there in rush hour, it could take me up to an hour or longer. So those are things that Trish, you said you plan for in terms of like
Yep.
going early, getting home before rush hour, all those types of things. So that's those are important things I just wanted to make sure that people understood about. I see another question about being treated at smaller centers versus larger centers, and if there's any kind of concerns there. I think you were both treated at a large center downtown, so that might not be something you can answer, but I think it's important to know that the care you receive as part of the trial would be the same across all centers, that what people are doing to adhere to the study and make sure that the science is credible would be the same no matter what center you're at. We also had a question about participating in a trial that's not close to you. And so I can share that I do know individuals who have, for example, participated in studies in the US. Sometimes the National Institutes of Health there will cover all of your expenses to fly there and participate in studies and stay, but that's not always the case. I've known people who participated in those studies, and then they've also paid out of pocket to participate in studies in the US as well. So those are just really things that you should ask when you're considering being in a study and seeing how that might impact you. So I really want to thank both Joanne and Trish for sharing so much tonight. Before we close, I'm just going to ask both of you if you would just like to share anything else with the group that we haven't had a chance to talk about, that you wanted to leave them with in terms of your experiences or things you want them to consider if they're thinking about clinical trials. And then we'll wrap our part of the session up.
Okay, well, maybe I'll go first, Trish. I do think that it's important for people to consider clinical trials and to, once you have a disease that has that doesn't have a cure and has ongoing research, it's good to start to get to know some of that research. I know it can be difficult if you don't have a medical or biological or background, but you can start by asking questions of your healthcare providers. And I've, it's not just the doctors you need to talk to, but also the nurses. They have a lot of information. And I know at Princess Margaret, they also have a library that you can access some of the materials. So it's, it's important to be your own advocate within your healthcare. And so it really helps to ask questions and learn as much as possible about the disease that you have. I know that one of the first things that I did was to join a couple of Facebook support groups for ovarian cancer. And sometimes I just, it just is overwhelming to read some of the things in there.
Right.
So sometimes I just kind of don't look there. But I have learned some things from some of the people there. And that's been helpful in my progress with learning about my own ovarian cancer and what it means to me. So I would encourage everybody to become, to improve your health literacy about your own health care. And you might find that you actually get better care when you understand it better.
Thanks, Joanne. Those are wise words. Trish?
To me, I just want to say thank you to, getting cancer. I guess people always shake their head at me, but it really has opened my eyes to a whole different world, and a world I'm now want to advocate more for research, trials, and ovarian cancer. Like I want teal to be the #1 color, not the other ones. Not that they're not important, but this has hit us, and I have three boys too, so and a grandson, and they're all behind me. So it's something that I'm testing the waters with, and want to, like I said, I can't thank enough for Princess Margaret and for that team. And reading about it, I have joined, like Joanne said, different Facebook pages, but a lot of it's in the States, so it's different. But having the ovarian Canada cancer and some of this stuff tonight that I didn't know about. So to me, this is a learning night for me too. It's wonderful. I can't say thank you enough to all of you. So.
Awesome. Thank you, Trish. Thank you, Joanne. It's been my privilege to spend the last 45 minutes with you. And thanks to the audience for all of the amazing questions. And I'm going to wrap up and pass back to Ovarian Cancer Canada. Have a good night, everyone.
Thank you, Dawn, for facilitating the discussion.
Thank you so much to Dawn, Trish, and Joanne for sharing all of those insights with us. And I'm just going to be here for just about 2 minutes just to make sure that everyone's aware of a new tool that we have available. It's a self-advocacy tool. So I just want to talk about that just very briefly. So a key pillar of our strategic plan is to improve care for those diagnosed with ovarian cancer. So to do so, we're developing a whole group of tools that are going to enable people living with ovarian cancer to make informed decisions about their care and just to ensure that they receive quality, relevant information in a timely manner. So that said, the first tool we've developed this year, and we're so excited to share with you this evening, is our clinical trials tool. So I've seen some conversation in the chat about this, but just to make sure we're all aware, the Every Woman Study Canadian edition taught us that very few women from across Canada with ovarian cancer remember being provided with information about clinical trials, and then even fewer participated in clinical trials. And we also learned, as the conversation showed us, there's just so many commonly held misconceptions and myths about clinical trials. So these were extremely important findings, as we know that clinical trials can connect people with new and emerging treatments for ovarian cancer, and they're so essential for medical advancements. So therefore, we've developed resourcing to help clarify these myths and misconceptions that are commonly held, and to provide information to those who want to learn more about clinical trials and to maybe find a trial that's right for them. So the first part of this resource is what we're calling a talk to your doctor card. And I'm going to show this to you in just a moment. So this is a printable card, or you can download it to a cell phone, and people can take it with them to their doctor, and it can facilitate a conversation about clinical trials. So it offers a brief definition of clinical trials, some important facts, and then it outlines some important questions you could ask your doctor. And then there's also space for you to take notes. And then to support this talk to your doctor card, we've also launched a new page on our website. So ovariancancer.org/clinical trials. And I'll show this to you in a second as well. And here there's just so much more in-depth information about clinical trials. All of the information has been divided into 4 sections. So clinical trials, the basics, finding a clinical trial, deciding if a clinical trial is right for you, and participating in a clinical trial. So that all said, I wanted to say 2 big thank yous. One is to Dr. Dawn Richards at Clinical Trials Ontario, who we just heard from. So Dawn shared with me a CTO's toolkit for organizations like ours to help us communicate high quality information about clinical trials to our community. So a lot of the information that we used in the development of our resources was informed by Dawn's work. So just a huge thank you to Dawn. And really, so most importantly, we want to thank our outstanding patient advisory committee that advised on the development of this tool, just to ensure that everything that we're sharing with our community are informed by patient expertise and insights. So we had a really fabulous group of 8 women with ovarian cancer. So in no particular order, I'd like to thank our fantastic committee members for their time, their dedication, their knowledge, everything. So Julie Pauling, Julie Mulligan, Livia Uy, Patty Pitts, Donna Pepin, Peggy Pickett, Erin Barrett, and Martha Hoyt. So I'm just going to share my screen in just a second so that you can actually see where these clinical trial tools live on our website, and then I will stop talking and we can move on. So here is my screen, and as you'll see at the top, this is ovariancanada.org/clinical trials. And here is where all that information lives about clinical trials. So you can click on each of these to drop down to learn more. And a lot of the questions that came up tonight in the conversation are covered on this page. And then here is where the talk to your doctor card lives. So you can click on this. You can see the card. So some facts, some questions you might want to ask, and some areas to take notes. So you could print that off and take it to your doctor and hopefully have a really good and meaningful conversation about clinical trials. So that's all for me, and I'm just going to pass it back so we can move on to the next session. And I just so you know, I'm going to put my contact information in the chat. So if anyone has any questions about the information available in the tools, you can contact me directly and I can help support you through all that. So take care and thank you, and I hope you enjoy the rest of your evening.
Thank you, Allison, for sharing that amazing tool that you've created. And I also just want to say, wow, that was an amazing and powerful session. Thank you so much, Trish and Joanne, for sharing your experiences with us. I know from reading in the chat that everyone here appreciates it as much as we do. Dawn, thank you too for sharing your expertise and being with us tonight and answering some very common questions and myths surrounding clinical trials. So thank you. Ovarian Cancer Canada is building research capacity as well as unlocking insights into rarer forms of the disease. I would now like to introduce you to Dr. Anna Tinker, who will speak to us today on the treatment of low-grade serous ovarian cancer and why one size doesn't fit all. Dr. Tinker is a medical oncologist at BC Cancer and a clinical associate professor in the Department of Medicine at the University of British Columbia. Her clinical expertise is in the treatment of women with gynecologic cancers. Her research interests are in the area of clinical development, immunotherapy, and translational research. Dr. Tinker will be interviewed this evening by 2 members of our patient partners and research team, Donna Pepin from Barrie, Ontario, and Erin Barrett from Vancouver, British Columbia. So thank you all for joining us, and I'm going to pass it over to you, Dr. Tinker.
Hi, good evening, everyone. It's a true honor for me to be here. Thank you very much for inviting me. I really look forward to our discussion. I think you've heard my introduction. I treat women with gynecologic cancers as my primary clinical focus. And I've been asked to speak to you folks today about low-grade serous ovarian cancer. And then, we'll engage in a discussion. I think if I'm correct, and if it's okay for me to take the lead, I meant to give you a little bit of a very quick overview of what low-grade serous ovarian cancer is, or what we understand so far. And it's very brief, just a few slides. And that will hopefully set the tone for, some of the discussion we're going to have coming up. So I'm just going to try to share my screen. And I apologize if I look off to the side. I have two screens, so it's just a little easier if I have things on two different screens. Okay, so what are you seeing now? I hope you're seeing a screen that says low-grade serous cancer of the ovary.
Yes, we can see
My
it.
opening slide. Okay, perfect. Thank you. Okay, so let's just talk a little bit about low-grade serous cancers. So as you all know, and I know I'm speaking to a very well-educated crowd, we have multiple ovarian cancer subtypes, and understanding this has really shaped how we approach patient care. Low-grade serous cancers are one of the 5 most common subtypes, but they are amongst the least common of those 5, representing roughly 5% of ovarian cancer diagnoses. They sometimes have a precursor lesion. They can arise out of a more benign change called a serous borderline tumor, but not always. And as far as we know, this type of cancer is not associated with hereditary syndromes, which is good for families. But oddly enough, it does affect women who are slightly younger in their age, which is something we see more often with hereditary syndromes. Nonetheless, this is not usually inherited. And as we study cancers more and more, and we study all these various molecular and histological subtypes, we realize that low-grade serous cancers have their own special mutations. KRAS and BRAF are some of the common ones we talk about, but there are other mutations being identified. And knowing that is important because that means that we know of a pathway that might be active in these cancers. And this pathway can sometimes be targeted by specific treatments. In our case, we're talking about something called MEK inhibitors. So this is another representation of the sort of the frequency of low-grade serous cancer as being very low frequency. And important in this as well is that unlike the more common type of serous cancers called the high-grade serous type, these tumors don't usually have a P53 mutation. They're not associated with BRCA1 and 2 mutations. They have a very different look to them under the microscope. They don't look very aggressive. They sometimes have these little calcium deposits, which you see here as little dark dots, which tell the pathologist that this might be a low-grade type of slower growing cancer. So the patient characteristics, as I mentioned, are often women who are diagnosed at a younger age. It's really important because it can affect women during their productive years of life when they might be young mothers. They're still in the workforce. They might be looking after their own parents, and then they are affected with a cancer like this. Although it's low-grade serous cancer, it is actually very often diagnosed in an advanced stage. So it's quite common to see it having spread already inside the abdominal cavity, much like we see with the high-grade variant. And although most women with low-grade serous cancers tend to live longer than their high-grade counterparts, we do see women whose disease can progress quickly and can be very difficult to treat. And definitely it is a very serious and life-threatening disease for many women who are diagnosed with it. As we study these cancers more and more, we realize that nothing is as simple as just seeing 1 or 2 different types of genetic changes. My colleague, Dr. Mark Carey, who works closely with us in BC and is well known for his research in low-grade serous cancers, has started modeling and studying a low-grade serous disease and looking at the different type of molecular groups that might be identified. And sure enough, there's more there than just knowing that it's not a hereditary cancer or that it expresses KRAS or has a KRAS mutation, or that estrogen receptor is often present, which is a feature of this disease. It very often expresses estrogen receptor. So now that we know there might be a few different types of groupings of this disease, it influences how we might think about it going forward, and we might have to increase our sophistication as we research this disease further. So when we think of treatment, as for all gynecologic cancers, of course, good surgery is important. And we do want our patients to have the best surgery possible, hopefully with a gynecologic oncologist. Sometimes women will have multiple surgeries. Sometimes they will have a surgery, and if the cancer comes back, they might have a second surgery, and sometimes even a third if it's still felt that surgery can do a good job at removing the cancer. Chemotherapy is challenging with this disease, and this is a theme across cancer. Slower growing tumors do not respond as well to chemotherapy because chemotherapy mostly depends on DNA replication being disrupted. So if cells are not replicating very quickly, when we give chemo that goes into the system and then gets cleared out of the system, if the cells weren't replicating at that time, they may not be affected by the chemo. So the response to chemo is considered quite low for this disease. Some women will respond, there's no doubt. I don't want to say it's not useful, but it's certainly nowhere near the response we see with the fast growing serous cancers. And as I already mentioned, most low-grade serous cancers express the estrogen receptor. This is something seen in various cancer types, and breast cancer would be the perfect example of one of the well-known types of cancers that express estrogen receptor. And anti-estrogen is a treatment option. Now, getting the cancer to shrink with anti-estrogen treatment is not that easy, but the cancer can be very stable on anti-estrogen treatment. And down here, I've just put in some pictures of how the pathologist would study the cells to see if it expresses estrogen. And here you see this brown staining is an immunohistochemical test that the pathologist has applied to a low-grade serous cancer. And they can see that compared to, say, a negative control, this stains positive for estrogen receptors. So this is a common thing that we test for in low-grade serous cancers, and anti-estrogen treatment is a common strategy that we would use to treat this disease. So the National Comprehensive Cancer Network, which is basically an organization out of the United States, has pretty much created this treatment paradigm, and I don't want to go through it in great detail, but it gives some guidance over what to do in different stages of the disease. And you can see here chemotherapy for women with advanced stage disease, so stage 2, 3, and 4. Chemotherapy is an option, but also hormone therapy is an option, one or the other. And if a patient is given chemotherapy, they might go on to hormone therapy after. So hormone therapy is an important component of what we do for this disease. If women have progressed after their initial therapy, as I mentioned, surgery might be an option, but there's a new strategy now looking at something called MEK inhibitors. So remember I mentioned that KRAS is an often abnormal signaling pathway in these cells. And downstream from KRAS, I don't know if we can see KRAS that well in here, and I'm sorry, this is a busy little picture, is something called the MEK protein, which is a signaling protein that eventually down this pathway tells the cell time to grow. So if we can interfere downstream of the KRAS, RAS, RAF, MEK pathway, then we might be able to block these growth signaling pathways and get the cancer to slow down. So that's been studied with a drug called trametinib. It is a MEK inhibitor, works right here by blocking this pathway. And it is a good drug in the sense that we see patients will respond to it. And many, many patients have stable disease when they take this drug. And we see that the survival of patients is improved when a trial was done comparing this MEK inhibitor against a physician choice of treatment. Unfortunately, the treatment has side effects, and probably between 25 and 35% of women might come off of it due to side effects. We've designed a trial, and this is a trial that's garnered some support from Ovarian Cancer Canada, and I'm really thrilled and thankful for this. And this is something we're really hoping we will succeed in launching. It's going to take us a little bit of work, I think, but we are certainly moving towards that. And this is a trial where we actually want to look at targeting both the estrogen receptor and this MEK pathway together. So that's not been done before. And there's actually some other scientific rationale for doing this. So people have looked at this in cancer models and they see that it might have an impact. And we want to give patients a MEK inhibitor, and we want to give them something called a serum, I'm forgetting what the S stands for, selective estrogen receptor degrader, which is a new, even more powerful anti-estrogen treatment compared to, say, letrozole or other treatments we have. And we want to see how a MEK inhibitor compares to one of these new anti-estrogen treatments and how they also compare to when they're combined together. So we've been very, very fortunate to have the support of Ovarian Cancer Canada in trying to move this study forward. And this is something we're hoping we can bring to Canadian women as a clinical trial. I'm hoping I'm not being too optimistic, but hopefully this year we will be able to move things forward in this regard. So this is a quick overview of where we stand and what we're hoping to achieve here in Canada with low-grade serous cancers.
Dr. Tinker, thank you so much for being here today, and especially thank you for your research focus on low-grade serous cancer. Not only are you collaborating on groundbreaking research, you also work, as you've said, in the Gynae Oncology Clinic, caring for patients with ovarian cancer. I was diagnosed with ovarian cancer in 2006, and to say that treatment for ovarian cancer has changed in the last 15 years is an understatement.
Mm-hmm.
And as you've also just said, we now know that broad spectrum cytotoxic agents are typically not helpful in treating low-grade serous disease, and that we need molecularly targeted therapies. Can you tell us, please, how long you've been treating patients and how your treatment of low-grade serous disease has changed and improved over the years?
Yeah, thanks, Donna. Well, if I tell you how long I've been treating patients, you might figure out how old I am. No, I'm kidding. I've been working here at BC Cancer as an oncologist since 2006. Prior to starting here, though, I did do a research and clinical fellowship for a couple of years in the area of gynecologic cancers. So I've been at this for a little while now, approaching 20 years. I think I feel very lucky to have joined this field when I did, because everything started to change right around the time when I became an oncologist. We used to call all of these cancers ovarian cancer, and they to us previously were all just one disease, which is crazy to think of now, of course, because we've moved that paradigm's changed so much. But we now really care about these groupings, not only the histological, low grade, high grade, all that, but we really care about understanding the molecular profiles. We used to give a lot of chemotherapy to women with low-grade serous cancers, and really that's changed. We're trying to do all we can to avoid that. Hormone therapy as much as possible, and we really, really want the hormone therapy to work because it's the easiest to tolerate. Women feel well on it often, usually better than chemo, and it's often much safer. And certainly that's our first go-to. The MEK inhibitors are definitely now here. In Canada, they're not funded, but we do have access to them through the company that makes those drugs. So that's been a really nice opportunity for us with our patients. We do molecular profiling, testing of the tumor to see which genetic aberrations are there, as a very common thing that was not available. I mean, really, it wasn't even routinely available like 7 or 8 years ago. It's a very new change. So we're always looking at the tumor characteristics, trying to determine if there's something there we can find that will give us a unique opportunity that doesn't exist, before or otherwise. And of course, trials, right? We're trying our best to bring in these new options for women and record the results and hopefully share those results with others. Yeah.
Amazing and so encouraging for us low graders. And also one thing I wanted to ask you about, and I know that everyone who's interested in low-grade serous disease today will agree with me on this, and that is it's very confusing for patients knowing that the standard of care for low-grade serous disease varies widely, not only within Canada, but around the world. It seems like everybody's doing something different. I guess I can ask you, I don't know, would you like to say what's your standard of care, or can you speak to that?
Well, I think that reflects a couple of things. Number one, when you have a rare cancer, That's not as extensively studied as the more common cancers. People start to interpret things their own way or approach the disease based on smaller studies or little series, or even their own experience, obviously. I think because we don't have that abundance of data that we might have for the other cancers we treat, it does give us more wiggle room. And that means there might be several very reasonable options and several ways that are, right, or at least not wrong, right? And there's going to be a lot of personal bias that comes into that. So for example, we work with our American colleagues a fair bit. They very strongly still believe that chemo should be an integral part of the first line treatment of low-grade serous cancers. Now that might not be uniform, but of the vocal people, you hear that. And you might still hear that a bit in Canada too. Others from other regions of the world might feel less strongly about that, and emphasize the anti-estrogen treatments more. It's, for me, I think it's a discussion with the patient, right? It always comes back to the person sitting in front of you and their profile and what their personal preferences are and desires and what are their immediate needs. Are they in severe pain and needing urgent treatment? Are they actually quite well? They don't have a lot of cancer. There's some time to make decisions. And you talk about these things, you have to discuss with every patient, what does chemo look like? What are the chances of benefit? What do these other treatments look like? What are the chances of benefit? What are the risks of all of them? Can we start with one and sequence through all of them eventually if we need to? Yes, no, why not? It's tough when you don't have really solid data to tell you, but I think you have to be open to that discussion with your patient. And the other thing, and Donna and any low graders out there, you guys are often in touch with other people. So sometimes the patient comes and says, hey, I heard of this. And you as an oncologist have to be open to hearing that and saying, yeah, okay, let's look into it. Is that good? Is it crazy? Can we do it? Should we? What's the risk? What's the benefit? Every patient is different and the decision process has to be followed. Yeah.
Thank you. It sounds from everything you've just said that it's, I shouldn't say confusing, but it's as challenging for you as a physician to know what the best path forward is when not a lot is understood. And thank you for considering everyone's individual priority. And Erin has a question.
Sure.
Thanks, Donna. Hi, Dr. Tinker. Thanks for giving us your time today. I really appreciate it.
Of course.
Well, I'm not a low-grade serous lady. I am of a rare subtype as well. I had a mucinous carcinoma. So I appreciate the fact that people like you, researchers like you, are investing your time in these more rare diseases. So could you take us through perhaps what types of challenges you face in researching a rare disease like ours and why there seem to be so few clinical trials available for low-grade serous cancers?
Yeah, I mean, some of the challenges, I guess, are self-evident. So, when you do research, the best or most reliable research is usually done in large numbers of patients. It's often the research might be like, for example, a clinical trial that we might expect to change care. That clinical trial would have to introduce a new therapy, but compare it to something old. And the way our systems work is we would expect the new therapy has to be proven to be better than something that we already have, because otherwise it's not viewed to be of any advantage to use it. So when large studies are done with large numbers of people, we can randomize some to the new treatment, some to the former treatment, compare them statistically, and we get a lot of clever people to tell us how many patients we need to be certain that difference we may be looking for is observed and is meaningful, and that we can be sure that it's a real difference, not just a random observation. That requires often hundreds of patients. And that with the rare cancers, that's only 5% of all the ovarian cancers we see in Canada. It becomes much harder to do those kind of trials. Not all patients are eligible for trials. Not all patients live in regions where they can come to a center for trials. So all sorts of things start to diminish, there's an attrition, how many people would actually be able to participate. And so there's just that barrier of having a rare disease and not being able to get high numbers of people to participate. That's one. The sad truth is, and I think probably the people on this call all know this, the people who have new drugs and money are usually our industry partners. Now, industry partners, companies that have these drugs, they do lots and lots of research. They really do try to bring a lot of good opportunities to our patients. The MEK inhibitor study that changed care was one of those examples. But when there's a really rare cancer, some partners may not choose to invest because they are also a business and they do not maybe always see how that will be a return for them in terms of their investment. Some of them will do it because they're, it's one of their company mottos to support rare cancer research, but some are less invested or willing. Sometimes drugs are going off patent. That's a major problem. If a drug is leaving it, the patent is about to expire, the company very seldom engages in a lot of research because they're now moving on to their next agent. They've done their development with a drug and no longer wish to invest. So it can be very challenging from those fronts. Even granting agencies, like we have several granting agencies in Canada, will sometimes acknowledge like this is a very important study that you are interested in doing, but is it feasible? Because it's a rare disease. Will you answer the question? I'm very grateful that Ovarian Cancer Canada has picked up our study as one to conduct, still acknowledging that we may or may not be able to complete this study. It will really depend on how lucky we are with getting it launched and successfully completed.
Thank you so much, Dawn. I'll pass it back to you.
Sorry, Erin. Thank you. And thank goodness for Ovarian Cancer Canada making research a priority for the rare types of our disease so that every woman has a chance. Dr. Tinker, your research with Dr. Mark Carey has shown that there are distinct types of low-grade serous disease. Who knew?
Yeah.
In other words, your data has revealed unique molecular subtypes. We now know that every low-grade serous patient has potentially a different cancer from the lady beside them.
Yeah.
Individual subtypes behave and respond differently, which explains a lot about why outcomes can vary so widely in patients with the same disease. Your research goal alongside of Dr. Carey is to establish biomarkers, being a test to predict who will or who will not benefit from a drug, drugs like MEK inhibitors or anti-estrogen therapies.
Yeah.
So this would represent an amazing scientific advance. So can you please explain more to us about how biomarkers could help to guide the best treatment choices for patients?
Yeah, thank you, Donna. Biomarkers are a really hot topic in oncology research right now. So, alongside that trial that I've shown you, the one that we hope to launch with the support of Ovarian Cancer Canada, we intend to collect a lot of tissue samples from patients, archived, like those that are already stored in pathology labs, and also new biopsies. And we want to apply this new molecular classification that Dr. Carey's lab has identified to every participant in the trial to see which of those groups they fit into. And then what we want to do, if we're successful with what we've set out to do, is we want to correlate whether responses to our treatment are correlated to any specific one of these profiles, right? And the importance of that would be, although our study is quite small, it would be one of the first steps in trying to identify what tumor features actually predict the kind of response a patient might have to therapy. And that's a biomarker. A feature, usually a tumor feature biomarkers can be anything, but in this case, it would be a genetic feature of the tumor that predicts whether a response will be seen or not. If we can see some correlation between those baseline features and treatment outcomes, that will really let us develop future studies that are much more intelligent so that we can we can enrich our patient population for those patients that we think are most likely to respond to a specific treatment. So, if we can tell that patients with a certain genetic change never respond to a MEK inhibitor, that would be helpful, because why would you put a patient on a toxic drug that 30% of women can't tolerate? We would avoid the toxicity, and maybe we would be able to tell that their tumor is very, very specifically sensitive to pathways that target the estrogen pathway, or drugs that target the estrogen pathway. So we might choose to study new drugs that target the estrogen pathway instead and put them on that. I see a quick note that came up that's very promising. That is truly our hope, is to stop subjecting women to random treatments because we can't pick who responds and who doesn't. And really try to be a lot more intelligent and say, look, we see this in your tumor. 90% of women with this change do well on this treatment. We want you to have this treatment. That's the future we're heading towards. So this is the work that will start that process. What's really great for the group I'm working with, and the credit goes to Dr. Carey because he's just been tireless at this. He's working with a lot of international collaborators, all of whom are really interested in this disease. So the findings that we make here, I think, will translate very quickly into a larger research setting amongst other countries and other collaborators. And we might be able to accelerate, these discoveries into research and trials around the world. So it's exciting. I really want to see this type of opportunity, this type of trial go ahead. Yeah.
Amazing. Thank you.
That's so inspiring to hear, Dr. Tinker, that the work done,
Wow.
partially funded by Ovarian Cancer Canada, will be translated so quickly to other women and other patients around the world. That's really wonderful to think of our Teal Sisters globally. Following along the lines of what we were just discussing with Donna, can you explain a little bit about how you and your colleagues are rethinking phase 1 trials? So taking on this note of, instead of making patients sick, making the cancer sick, and reducing toxicity by reducing doses and combining drugs?
Yes. Well, yeah, there's a lot of interest in this. A lot of these drugs that are not the conventional chemotherapy drugs, there is a difference between what we call, in chemo, we used to find the maximum tolerated dose. We would try to get to the highest dose that is safe to give to a patient. But with some of these drugs, there's a disconnect between what is the maximum tolerated dose and what is the effective dose. So that's one thing that's very important. And then the other thing is, sometimes there are interesting synergies. So you might combine drugs that work in slightly different pathways, but those pathways are both important for how a cancer will respond to treatment. And by making the combination happen, you need much less of both drugs. Whereas if you only used one drug, you would still have to go with the higher dose. So these are really wonderful sort of strategies to try to give less toxic treatment, but effective treatment. And of course, the perfect scenario would be is if we had a biomarker to tell you, this is the treatment you should have. You can have it at low dose because we know these drugs work well together. That would be ideal. Yeah.
Thank you. I think we are way over time on our session, unfortunately, and I have so many questions that I still wanted to ask you. But as a low-grade serous patient myself, and on behalf of my community, I just want to say thank you, Anna, for focusing on our disease as a separate entity. And is there anything else that we haven't asked you that you would like to say?
Off the top of my head, only just really want to thank, all of the participants, women, advocates, Ovarian Cancer Canada, who have just worked so tirelessly to make, these wonderful opportunities happen, and for sharing our message, and, just reaching out and making sure that all the women who have this disease and other ovarian cancers are well informed and connected and speaking to each other. So thank you all. Yeah.
Thank you.
Thank you so much.
Dr. Tinker, I wanted to thank you too for your overview and explaining low-grade serous carcinoma in words that I could understand. And also for your expertise and honesty in answering some really tough questions. So I appreciate that. And Donna and Aaron, thank you both as well for taking the time this evening to share your knowledge and experiences with us. As part of the audience, I know it's really incredible to be able to learn from lived experiences like your own. So thank you both. So now that wraps up the first half of our evening. So we're running a few minutes late, so we're going to have a 10-minute break. I would encourage you to get up and have a stretch, and we'll meet you back here in 10 minutes. It's on mute there, sorry. Welcome back, everyone. We are going to jump right into the second half of our evening tonight. And the first session up is the PARP Revolution, how PARP inhibitors are changing the landscape of ovarian cancer treatment. I'm excited to introduce you tonight to Dr. Shaina Lee. Dr. Shaina Lee is a gynecologic oncologist at the Alan Blair Cancer Center in Regina, Saskatchewan. And tonight, Dr. Lee will share with us some of the latest information on PARP inhibitors and how they're changing the treatment landscape. Dr. Lee will be interviewed tonight by two members of Ovarian Cancer Canada's Patient Partners and Research Team, Peggy Pickett, who is a Teal sister from Niagara Falls, Ontario, and Patty Pitts, who is from Vancouver, BC. Patty lost two close friends to ovarian cancer in 2018 and has been a member of the Patient Partners and Research Team since its inception. So thank you to all of you for joining us this evening. And Dr. Lee, I will pass it over to you.
Hi, everyone. I'm Shaina Lee, and I'm a gyne-oncologist from Regina, Saskatchewan. So during this session, we will try to spend most of the time discussing and doing a Q&A session. So I will keep my presentation quite short and simple, so that we can have as much time as possible for our discussion. So I will just share my screen. All right. Okay. Can you all see my screen?
Yeah, we can see it, Shaina.
Okay. So yeah, we will spend the next 10 minutes or so going over some background information on PARP inhibitor in ovarian cancer. So we will talk about what a PARP inhibitor is and who's eligible to take a PARP inhibitor in Canada at the moment, and what types of PARP inhibitors are available here, and what kind of clinical benefits have been shown in previous trials. So I've had several patients who ask me how a PARP inhibitor works and why it will work well for those with BRCA mutation. So I thought I would try to explain the mechanism action of PARP inhibitor. So our human DNA has two strands, and as the cells are dividing, as we live, there can be some errors that are made in DNA when they're dividing. So those errors may be error in one of the strands or breaks in both of the strands. And we intrinsically have several mechanisms that can repair those damages on DNA. And PARP, or a poly-ADP ribose polymerase, is one of the protein molecules that can repair when there is a defect in one of the DNA single strands. So if we use something like a PARP inhibitor and inhibit the action of PARP, then the cell cannot repair the single-stranded breaks, and those breaks will accumulate and will result in double-stranded breaks. And if the tool to repair these double-stranded DNA is broken, then that cell might die. And we use that strategy in management of ovarian cancer because about half of the ovarian cancers are missing the tool to repair when there's a double-stranded DNA breaks. And an example of that tool would be homologous recombination. And those ovarian cancers that are missing those tools, we call them homologous recombination deficient. And BRCA1 or 2 associated ovarian cancer is an example of HRD tumor. So it'll make sense that if we give a PARP inhibitor to those cancers with HRD or BRCA1 or 2 mutation, then it'll likely lead to tumor cell death. So at this point, even though there are lots of research projects that are going on, trying to incorporate PARP inhibitor as a part of the mainstay treatment for ovarian cancer, it's currently used as a maintenance treatment for ovarian cancer in the advanced stage setting. The goal of maintenance treatment, we should remember, is to prevent or delay cancer recurrence and to prevent symptoms from cancer recurrence while maintaining excellent quality of life for patients, which means the treatment should have a goal of causing minimal side effects so that the women who are in remission is able to enjoy their time when they're off systemic chemotherapy. I wanted to make a note about this because sometimes we feel the need to be on a type of treatment, even though it may lead to side effects and worse quality of life. And we should remember that the goal should not be to make the quality of life worse. So who's eligible for PARP inhibitor in Canada at the moment? And I should also note that the eligibility criteria and medications that are available to us in Canada continue to change. So the information here now may be different a month or two from now, but this is the current eligibility information in Canada. So the first group will be those who have newly diagnosed advanced stage ovarian cancer, whether it is stage 3 or 4, once they complete their surgery and platinum-based chemotherapy. And the second group will be those with platinum-sensitive ovarian cancer recurrence, and they also have to have a completion of their platinum-based chemotherapy with or without surgery. And for both of these settings, the person must have a complete or partial response to platinum-based chemotherapy in current practice. There are two types of PARP inhibitors that are available in Canada. One is olaparib, which is provided for women with germline, another word for blood or inherited type of BRCA mutation, or somatic BRCA mutation, which is a tumor-specific BRCA mutation. And we use that for first-line and recurrent setting. Niraparib is offered to women with or without BRCA mutation in first-line or recurrent setting. And even though the combination treatment with olaparib with bevacizumab is something that's used in the States and Europe, it is not yet used in Canada. And I should also note that you all may know, but all newly diagnosed advanced ovarian cancer that are non-mucinous type, so examples will be high-grade serous or endometrioid type, should have their tumor and their blood tested for BRCA, because it'll change our counseling on how the PARP inhibitor will work for them, how beneficial it'll be for them, and the type of PARP inhibitor that they'll be able to receive. So in terms of duration of treatment with PARP inhibitor, the recommendation is given based on the trial result. So in the first-line setting, olaparib may be used up to 2 years, but can be continued if there's evidence of partial response. And typically, a person may be treated until there's evidence of disease progression. For niraparib, based on the trial regimen, it will likely be used for 3 years or until there's a disease progression. In the recurrent setting, based on the trial result, again, it will be used until there is unacceptable toxicity or disease progression. So how much of a benefit can one expect from PARP inhibitor? I think would be an important question that one would ask if their oncologists offer them with PARP inhibitor after completion of their platinum-based chemotherapy. So in the first-line setting, in terms of olaparib, the trial that showed evidence for olaparib only included BRCA mutation carriers, and the trial's name was SOLO-1. And based on this trial, where women with advanced ovarian cancer who had BRCA mutation were offered olaparib versus sugar pill, the time until disease progression or their first recurrence was 56 months for those who had olaparib versus 13.8 months for those who had a sugar pill. So the difference was quite significant at 42.2 months. So that was over 3 years of a difference until the time of first recurrence. And in other words, this would be in other words, the risk of death or progression was 67% lower with olaparib. And the overall survival data, the data on how long did they overall live, is the data is still not mature. So we're waiting for the final data. In the first-line setting for niraparib, we get the data from the trial called PRIMA. The numbers that you will see here will look different from what you just saw from SOLO 1 trial because they pre-selected patients who are ultra high risk in terms of the high-grade serous cancer. So they chose women who had residual disease after surgery or those who had stage 4 disease. And in this trial, they included all women regardless of their BRCA mutation status. So here, for all comers, the time until disease progression had about 5.6 months of a difference, whether they were on niraparib versus sugar pill or placebo. And in this trial, when they divided patients into their BRCA mutation status, those who had BRCA mutation had 11.2 months benefit by being on niraparib. And when they looked at patients who had that HRD status, the ones who are missing that the tool to repair the DNA damage, their outcome was quite similar to the BRCA mutation carrier, which makes sense because these HRD tumor carriers, even though they don't have BRCA mutation, they probably have other types of gene changes that result in HRD. So their outcome was 11.5 months of a benefit on niraparib. And in this trial, when they looked at women who did not have BRCA mutation and who did not have HRD, the benefit that they saw from being on niraparib was 2.7 months, which is the shortest. And I should note that the HRD testing of the tumor is not yet widely available in Canada. So that's the reason why niraparib is offered to women regardless of their HRD or BRCA status. And there will be some trials and a hospital or cancer center specific HRD testing that will become available in the near future. In the recurrent setting for olaparib, we get the data from a trial called SOLO 2. So very similar to SOLO 1, they only included women who had BRCA mutation. And in this trial, the difference in time until their next progression was 13.6 months with olaparib. And this trial has been available for a longer period. So they do have an overall survival data. And what they found was for BRCA mutation carriers who had a recurrent ovarian cancer, being on olaparib improved their overall survival by about a year. For niraparib, the data comes from a trial called NOVA trial. And this trial included all comers regardless of their BRCA status. In this trial, time until their next progression or recurrence was such as this. So for BRCA mutation carriers, there was a 15.5 months benefit by being on niraparib. For those without BRCA mutation, there was approximately 5.4 months of a delay in their disease recurrence. So based on this trial, the niraparib became available to those with or without BRCA mutation. Very recently, there was information that was released by the manufacturer of niraparib, as there was some updated information on overall survival data. This is not yet published in a journal. So based on some of the newer findings, they found that there was actually no difference in overall survival in the BRCA mutation group. So even though niraparib was resulted in delay in the disease recurrence, overall length of the life that a woman had on niraparib was not different when it was compared to those who were on placebo. And there was some trend for possibly a worse survival in non-BRCA mutation carriers. And because there was a trend, so trend meaning it was not statistically very distinct difference in survival, but there was some trend. So they're looking at this data very cautiously, and more information is expected in the future. And because of this, you may have some of the oncologists including this as part of the counseling when someone is, offered niraparib, and in the end, the decision to start niraparib versus not, or stopping in the middle, may be made after the oncologist has a discussion with the patient. So, so some of the adverse events and side effects that are expected from PARP inhibitors, these are some things that we saw from the trial and also from our daily practice. So some nausea, change in bowel pattern is expected, but it is fairly well managed with counseling and medication use. Fatigue is quite common. And some blood test results. So hemoglobin, white counts, and platelets may go down. And that's why we do quite close blood work follow-ups. And with the use of niraparib, high blood pressure or palpitations can occur. And there's a small chance of about 1% of developing acute myeloid leukemia or myeloid dysplastic syndrome from a PARP inhibitor. And as I mentioned, we do monthly blood work, and we do monthly or as needed assessment with the patients on PARP inhibitors. And based on the trials and our clinical experience, most of the side effects are manageable with interrupting the dose for a few days or modifying the dose. And the quality of life was maintained in all of the trials. And we also find that patients actually tolerate the medication very, very well. So these 4 objectives were what we just went through, and I will now move on to Q&A session. And I think we have some patient partners who will talk with us today. I will stop sharing.
Hi, okay, I'm Patty Pitts. Thank you, Dr. Lee, for your presentation. As Stephanie said, I'm the one member of the patient partners who isn't an ovarian cancer patient, but did lose, sadly, a couple of very, very dear friends to the disease. And I was quite involved in their care and support, and saw the side effects that they endured through their regular chemo treatments and other drug treatments. Neither of them, this was a few years ago, so none of them were eligible, even if PARP inhibitors were available back then. I'm not sure that they were. You mentioned that the main aim, of course, is to improve people's quality of life and extend their life. And you presented, there's a lot of information in your presentation that covered an awful lot of different scenarios, some very encouraging, others less so. But overall, can you tell us a bit more about your experience and with managing the side effects? You said that most of them are well managed, and that for the most part, people maintained a pretty high quality of life. But can you maybe expand upon that a little bit, of some of the side effects that people taking PARP inhibitors might be expected to encounter?
Yeah, I think that there are like different categories of side effects that we notice and we counsel them about. So based on the trial results, we do know that there will be some GI side effects. So I think the most important part was counseling patients prior to them starting the medication. So at our center, we give patients different options for anti-nausea medications available with them. And I usually advise my patients to take their medication, especially the niraparib, which is once a day, in the evening with some gravel if they wanted to try with gravel, because I find that like it really helps them not experiencing any nausea if they take it with some gravel in the evening. And I think that kind of knowing that this could happen is also a very helpful tool, because without proper counseling, some of the side effects can come as a surprise for patients. So I find the nausea is quite well managed with some medications. And palpitations and high blood pressure, we ask patients to monitor their blood pressure quite closely weekly for the first 2 months. And I've had to start some patients on blood pressure medications. And with that, I never had to discontinue anyone because of blood pressure issues, because I find that it's quite well manageable. And with the low platelets and hemoglobin, the reason why it can be difficult in the beginning is because they just finished their chemotherapy. So they're still recovering from their treatment. So it does require some time until their bone marrow and their self are both fully recovered from chemotherapy. So in those scenarios, we may reduce the dose of a drug in the beginning, and we may gradually increase as their tolerance improves, or we may do alternating doses of low dose and a higher dose. And eventually we will try to go up to a moderate dose for our patients. So I think those are some of the strategies that I use with my patients. And I find that most of my patients are tolerating it quite well at the moment.
Oh, that's excellent. Thank you very much for sharing that. There's, I'm not sure if I can do this, but I'm just going to pop over into one of the comments in case you haven't said it. Said, one of the participants said, very curious why placebo was offered in this case. Is it the stage of the trial? Seems that a lot of women's survival was put at risk. I don't, can you respond to that? Just you don't have time to get to it later on.
Yeah, I'm not sure which trial. Yeah, I don't know when that question popped up.
Oh,
I can tell you that with all the trials that I discussed, placebo was used, and after and the placebo was used because whenever we do a clinical trial to determine whether a new drug is effective, we either compare it to the standard treatment. And at that stage, the standard treatment for maintenance treatment was not being on anything. That's why a sugar pill, which is a placebo versus maintenance pill, was looked at. And in other, clinical scenarios, if we are comparing adding PARP inhibitor to certain medication is beneficial or not, then in that scenario, the patient will be receiving the standard treatment plus placebo versus standard treatment plus PARP inhibitor.
Okay, and actually my mistake, I think I was reading something was one of the previous presentations, so my mistake, but
no worries.
That was interesting to know and learn. Okay, over to you, Peggy.
Actually, an excellent lead-in, because I actually was on a clinical trial for a PARP inhibitor, and that was my first experience with a PARP medication. So I was diagnosed with high-grade serous ovarian cancer, stage 3C, in January of 2014. So I am now 8.5 years into this journey. And so 8 years ago, at the end of my treatment, I was offered the opportunity to be part of the SOLO-1 trial for olaparib, which is now trade name Lynparza. So I just briefly, like the experience of being on a clinical trial, I jumped on it because I think that we need to move science forward. I jumped on it because I believe that if I didn't get benefits, somebody else would. But probably 95% of my reason was that I saw my oncologist every month. I had, greater testing and follow-up and so on. So, so the clinical trial I was on was a double blind. I was offered either the medication or the placebo. It randomized. I didn't know which I was on. And at that time, there was no standard of care. So it was either you got the medication or the placebo. And I was on the clinical trial for the full 2 years. One of the hardest days of my cancer life was when they said I had to stop, even though I thought I was on the placebo. So my question is, and you covered it a bit in your presentation, but honestly, I got distracted by the chat. So
I'm trying not to look at the chat right now.
Right.
I don't want to miss any question.
Exactly. Well, so I put the chat away and I figured somebody else will point out if we have a question later that we have to. But anyway, so my question is, why did I have to stop taking that PARP inhibitor after 2 years when I was still no evidence of disease? I know it was part of a clinical trial, but I think you said that with olaparib, you still come off after 2 years.
So I think Dr. Drew and Dr. Tinker are on the line too, and they may be able to add. I believe the Solo 1 and Solo 2 like regimen also came from other trials that used it. And I wondered also another reason, maybe, if whether there is a continuous benefit even though you're not on the PARP at that point. And if there's a small risk of an AML, like is the risk for AML MDS, like that risk, does it trump the potential benefit that women will continue to have from PARP inhibitor? However, the recent update from the Solo did show that the risk of AML and MDS does not increase, even though even at like 4 or 5 years.
Yeah, but did you say the risk for that was only 1%?
Right.
And my risk of recurrence is way greater than 1%. So that doesn't equate to me. The fact that I have to come off because the risk of MDS, and whatever those things were, AML, if that risk is 1% and my risk of recurrence is extremely elevated over 1%, that makes no sense to me that's the reason that I would have to stop.
Yeah, I want to hear from Dr. Tinker and Dr. Drew, because I do get asked the question a lot, and I have a few patients on olaparib who's like kind of dreading that 2-year mark. But at that point, I'm planning to talk to them and then make a decision together. Because it is true, like in the first line setting, that's kind of the best time for us to have a control of the disease.
And I know my case was a trial, but I know that now it's approved. So what's the answer?
Hi, Peggy. Perhaps I can add to that. So I think the main thing is when we design trials, we have to decide what the trial is going to be. And obviously at the time that Solo 1 was designed, nobody knew how long to give a PARP inhibitor for to women who potentially were going to be cured of their cancer. And the trial could have looked at 6 months, it could have looked at 12 months, it could have looked at 2 years. And like Prima, it could have looked at 3 years. So actually the answer is we didn't know how many years to give these drugs for. So like in breast cancer, where you have, tamoxifen for a certain number of years, it's a similar thing. We didn't know how many years to do. So most women in Solo 1 didn't have what's called measurable disease. So you couldn't measure their response to treatment. So after 2 years, most women came off the drug because they'd completed the 24 months. And what's encouraging, if you look at the 5-year follow-up data for Solo 1, is that the majority of those women are still in response, even though they've had 3 years off drug. So that is encouraging. And you could argue that maybe 1 year is enough to take these drugs. And the AML, the acute myeloid leukemia risk, obviously at the time wasn't known. And so it could have been like 20% or 30%, and then you might be killing people of something else by curing another cancer. So obviously we have to be cautious. And I recommend most of my patients do stop at 2 years because of the risk of potential side effects and effects on quality of life. And there is data now, a 5-year follow-up for Solo 1 showing that a lot of women still are benefiting even though they've stopped treatment. But I think if you've got disease that can be measured, you know, on scans, then it is reasonable to think about continuing, but you'd have to discuss that, you know, with your own oncologist, what's the pros and cons of taking these drugs. And we don't know that having them indefinitely is the right thing to do, you know, in women who are in the first line setting, which is slightly different if you've kind of the cancer's come back. So I hope that makes a bit of sense.
It does. It does. And as a follow-up question, and I hope I see all these big brains on screen and I'm just going to capture them. As a follow-up question, I remained no evidence of disease for just about 2 years following coming off the trial. So 4 years to the day from my cancer diagnosis, I remained cancer-free, but 4 years to the day I received the diagnosis of recurrence. So believe me, I'm not complaining. I got 4 years and I've lived another 4.5 since then, right? So like, it's all good news, but I want it to be better. So I experienced a recurrence after stopping the PARP inhibitor. And I chose to go back into chemotherapy because I knew that olaparib had been approved because of the SOLO2 trial for a woman after recurrence if she responded to platinum-based therapy. So I chose that simply so I could get back on the olaparib. And I had to fight and to advocate for it. I had a really great team working on my behalf. And that did happen. So I was on the olaparib, had a recurrence about, I don't know, 18 months afterwards. Had radiation for the recurrence, went back on the Lynparza, and then had radiation again. So I'm now considered, I think, resistant to the PARP inhibitor. If I, you know, am in treatment currently, if I'm no evidence of disease after this, can I go back on olaparib? Can I go on to a different PARP inhibitor? Like for a woman who's been on a PARP inhibitor and has a recurrence, what are their options after that?
So
There's
I think
lots
that's
of us out there.
Yeah, and I think we'll be seeing more and more of women with similar situation. So for those who have a recurrence after being on PARP inhibitor, then that disease would be considered resistant to PARP inhibitor. And
After the first time on a PARP?
So no, like after someone is like, while somebody's on PARP inhibitor or soon after stopping PARP inhibitor, if they have a recurrence, then it would be considered resistant to PARP inhibitor. And we would think that, you know, they have developed different mutations that all of a sudden is making their BRCA functional, or perhaps they've developed a mechanism to, you know, kick out the medication from your cells so they are not functional anymore. So the consensus now is, you know, giving a PARP inhibitor again alone may not be a wise idea because it will unlikely work. The avenues that researchers are looking at right now for those who have a recurrence after trying a PARP inhibitor would be utilizing PARP inhibitor with other medications. So for example, women, there are trials that are going on, and what they would try to look would be combining PARP inhibitor with immunotherapy with or without bevacizumab, because there's an idea that, you know, if, you know, because ovarian cancer, so at the moment, evidence shows that using immunotherapy alone for ovarian cancer has not been very promising. But if we combine PARP inhibitor with immunotherapy, it may actually have some synergistic effect. And I think Dr. Drew was actually the author for the study called Mediola that used combination of PARP inhibitor with immunotherapy with VEGF inhibitors such as bevacizumab, and it showed that you know, regardless of their HRD status, there was some benefit. So I think the strategy for those who recurred after PARP inhibitor will be using a combination of PARP inhibitor with immunotherapy, with or without VEGF, and also other options that are available, although it's still a clinical trial setting, is there's a trial that's going on right now in several centers in Canada, including like Calgary, Edmonton, and Toronto, where a drug called antibody drug conjugate is used for patients with platinum-resistant ovarian cancer, because there was a single-arm trial that showed that for certain types of recurrent high-grade serous ovarian cancer, if we use antibody drug conjugate, again, it is a targeted treatment, there was a response rate as high as 30% or so. So there is a trial going on, and there will be a trial that's opening in the near future too, where chemotherapy with immunotherapy is used in comparison to single agent chemotherapy. So those are kind of trials that are actively happening. So, in the future, it may become a standard of care if we do see some good evidence. And also, I think someone talked about this earlier on the chat, but we still use the traditional single agent chemotherapy to people who have, fairly good kind of a quality of life, whether, how, depending on how symptomatic and how ill they are, we still do use single agent chemotherapy, which, does work for some women.
It's working for me right now. So,
yeah.
Right. I'm back on chemo with bevacizumab, and I'm responding. It's just, this is going to end. And I'm always looking to see, sometimes you just need to do something.
Yeah.
So anyway, so thank you, ladies, everybody, for answering my questions. Over to you, Patty.
Yeah, thanks, Peggy. And I'm keeping just a wee eye on the chat room, and this one actually is dealing with our session, Dr. Lee. Someone wants to know if you could repeat the name of the anti-nausea drug that you mentioned recommending to women who have that kind of side effect during their treatment.
So at our cancer center, for those who are on PARP inhibitor, we provide them with ondansetron or a Zofran, so that they can have it in the pocket. And then metoclopramide or Maxaran as another option. And then over-the-counter gravel. Some of my patients actually love taking Nirapur with gravel, and then they go to bed. It's so that's something that some people use. And, I always check them, like, are you still using the gravel? And then some of my patients said, I actually have zero nausea. So they just have it available just in case.
Great, thank you. And I just have one further question. The Ovarian Cancer Canada is really dedicated to believing that everybody should have access to the best treatment for their disease, no matter where they live in Canada. And as we know, that sometimes can be difficult. And so can you just sort of tell us what the situation is across the country for people wanting access to PARP inhibitors?
So my understanding is across Canada, but I think we did have a chat in the past that PEI may not have a PARP inhibitor readily available, but in most areas in Canada, PARP inhibitor will be available to those women that I described in the presentation.
Great.
It will be offered.
Okay, well, thank you so much, Dr. Lee. Now I guess we go over to you, Stephanie. Oops.
Muted, sorry. I'm going to jump in because the question and answer box here is on fire. So we have a lot of questions. We have about 7 minutes left, so we're going to get through as many of those as we can. So here's a question. My oncologist took me off niraparib last week as disease has returned. She advised that it's only showing to be effective on first-time disease patients, not for those who've had a recurrence. Can you comment on that?
So I think that likely occurred based on the recent release from GSK that showed that there may be some worse overall survival outcome for those who were on niraparib. I'm not sure why, if they actually had a good tolerance. I haven't stopped anybody based on that letter. So I think that this is something that you will see a huge variation in across the country. And I think that we're still waiting for more data to come out. I think Dr. Tinker and Dr. Drew both would like to comment. My plan moving forward is to share the limited information that we have available, and depending on the patient's, like how they're doing and what their disease status is like, I will continue to discuss the pros and cons of being on niraparib for those who are not, BRCA mutation carriers and who are in the recurrent setting. So I will let Dr. Tinker and Dr. Drew kind of give some comments.
Do you want me to go first? Yeah, I mean, I think for women who are having niraparib in the recurrent setting, so the cancer has come back, you had a round of chemo and then you're on the niraparib. So we're not talking about people who've had it after first line. Then you're generally on the drug until it stops working or you can't tolerate it. So there's no limit to how long you're on it for. And that should be still the main thrust of treatment. So if women are on niraparib and they are benefiting, so based on scans showing response, based on the CA125 being in response and being in tolerance of the treatment, then I do not think it's the right decision to take people off niraparib based on this new data that's come out from GSK. And that is because on an individual patient level, the benefit is the benefit that you're actually getting, not the averages from trials. And I think this new information from GSK, it's very important that we have new information. So we don't want to hide new information. And it's important that we disseminate this to our patients and it's disseminated to us as the oncologists, but then you have to interpret the information. So this new information is showing that the survival of women in that trial was slightly longer in those that got the placebo over those that got the niraparib. What we don't know is how many women in the trial came off placebo and went on to a PARP inhibitor. And what we don't know is what subsequent treatments those women had. Some of those women might have had better access to clinical trials and went into more clinical trials than those other women in the other arm. So overall survival is a difficult thing to show a benefit to in treatments where there will be lots more treatments coming afterwards, because those treatments coming afterwards affect your overall survival. So I think there is still maintained a benefit to niraparib, and women should still be on it if they're benefiting from it. But that decision really should be an individual oncologist and patient decision. And I don't think on this sort of kind of forum, we would be recommending individual sort of patients do something, but you should talk to your own oncologist. But I wouldn't be stopping. I agree entirely with Dr. Lee about I wouldn't be stopping women on niraparib because of this new data.
Yeah, thank you both. Dr. Tinker, did you want to weigh in?
Well, I'll just say I agree with that, and I've already started having some conversations with patients in this setting, it's really about sharing the information and deciding on an individual basis. But back to the question that was posed, I think what might have been said is it sounds like that in that particular case, somebody was on niraparib but then had progression. And in that case, I think that would mean that the treatment, the PARP inhibitor is no longer effective for that patient. And I would agree, although I don't know exactly the wording used by your oncologist, I would agree that right now, if you have progression on a PARP inhibitor, we don't really have any good evidence that going back on a PARP inhibitor, even a different PARP inhibitor, will have any benefit. And in fact, we really do think that olaparib, niraparib, rucaparib, there's a whole bunch of different PARP inhibitors in the world. We kind of, for now, think of them all as the same. They have a class effect. And if you've been on one, there's no known benefit to switching to another one. They're viewed to be equivalent. So if you've progressed on a PARP inhibitor, I think is probably not ever going to be a treatment you would go back on again. Peggy, you told us about your situation where you hadn't progressed, you stopped the 2 years. And it's a really interesting question about what will be the benefit for women who finish the treatment years later, have a recurrence. How much benefit will they have if we treat them with a PARP inhibitor? Again, that's actually a topic of some research we want to do and something we're proposing as a research study. But it's a really important question because there will be many women like you who complete their treatment course, will stay well for a period of time. And if the cancer comes back, do we give them a PARP inhibitor again or not? We need to know the answer. Yeah.
Thank you, Dr. Tinker. I have a couple, well, I have lots more questions, but I'm going to get to as many as I can here. There's a couple here with the same theme. I had to have my dose lowered several times down to 200 milligrams per day from the initial 600 milligrams per day. What kind of effect will that have on efficacy in the long term? Well, up to a 2-year horizon anyway, with the lower dose.
So that is a very good question. In the Solo 1 and 2 trial, they did have patients who had some dose reductions. My understanding is it's always a balance of keeping the medication on so that the patient can tolerate versus losing the efficacy. So the goal that we have is to try to increase the dose back again to higher than I think what did you say 100 milligram twice a day or 200 milligram twice a day, but I think it is a balance. I think that if I have a patient who's BRCA1 or 2 mutation carrier, and then if this is a frontline setting, I think that I will be trying my best to work with my patient to keep them on, even if it is a low dose, because I think that the benefit that we saw from Solo 1 is significant, and given that in the future I may not be able to put them back on PARP inhibitor because after they have a recurrence and their bone marrow is affected by the chemo again, I don't think that the tolerance will be better. Like now is the best time to try. So I think that the person will be having a conversation continuously with their oncologist to decide, but I think being on a dose of PARP will be better than nothing.
I think it's important as well, when we develop these drugs, we just kept increasing the dose to being able to tolerate them. So that's how drugs are developed. And we know that all PARP inhibitors are probably dosed too high, and most women need to have the dose, I don't call it dose reduction anymore, I call it dose corrected. So if you are on too much drug, then you're getting too many side effects and your bloods aren't okay, then you're on too much drug and we need to correct the dose. So we know there's data from the NOVA trial showing that whatever dose of niraparib you're on, it works. And it's because we make up these doses based on the early trials, and we just dose people 300, 200 or 100. But every patient is different in terms of their biology and their size. So for a lot of people, it's too high a dose. So I know patients, I know you get worried when we want to change the dose, but it's usually we're still giving you what's called therapeutic, so effective doses, and we would never go lower than what has been shown to be effective. And I think probably 2/3 of women need the dose reducing, and that's very common. So I started to call it dose correction now. I feel that that's a easier thing to discuss.
Yeah, and I'll note that based on the for the trial that used the niraparib, they started at a maximum dose of 300 milligram for everybody, and then they did notice that women had difficulty with tolerating. So then they actually made a different criteria. So people with lower body weight and low starting platelets, we would generally start them at a lower dose. And based on the trial, it did show the same efficacy. So I agree with Dr. Drew that it is a dose correction.
And I've just seen in the chat somebody said dose tailoring might be an even better term. So I think we've all learned something tonight.
Very nice. Thank you both for that. We have time for one more question for this session, but I just want to let everybody know that obviously this is a very popular topic, so we will revisit it at another time and be able to have another session on PARPs. But for now, we do have time for one more. If ever bevacizumab is approved in Canada and a patient is already on olaparib, Do you think it will be beneficial or even possible for one's doctor to add it to the olaparib treatment, even 1+ years onto the PARP treatment?
That is a very good question. So bevacizumab, as you may know, is already approved to be used in settings of advanced stage ovarian cancer in the first line setting and in the recurrent setting, whether it is mostly for platinum resistant setting. But given that we don't have a data of adding olaparib after, adding bevacizumab after being on olaparib alone for a year, I do not think that it'll be something that's offered, because we create standard of treatment based on clinical trials. So we will need a trial that uses that exact regimen in order for us to actually use the combination of bevacizumab and olaparib in the future. So I think what may change in the future may be, like continuing with bevacizumab and olaparib as soon as they're like in the first, like in the frontline setting after initial chemotherapy. But I don't think that we will be adding bevacizumab after somebody is on maintenance for a few years if there is no evidence of disease progression.
Anything to add, Dr. Drew or Dr. Tinker?
No, I agree with Dr. Lee. I think we have to follow the data and what the evidence, shows. I think, and that's important.
Okay, I want to thank you all for participating in this session. Dr. Lee, thank you again for helping us to understand some difficult subjects in your presentation at the beginning. Dr. Tinker and Dr. Drew, thank you also for jumping in to answer the ton of questions we had. Peggy and Patty, for your insights and your experiences and for sharing with us this evening. Thank you. As I mentioned, this is obviously a very popular topic, so don't worry, we will revisit it at a session in the future. And with that, I'm going to ask Dr. Drew to stay on because she's going to be presenting our last session of the evening, the NEOCATS study, a potential paradigm shift for first-line treatment of high-grade serous ovarian cancer. So Dr. Drew has played a key role in both the preclinical and clinical development of the PARP inhibitor rucaparib in ovarian cancer. Her research interests include the targeting of DNA repair pathways as treatment for gynecological cancers and PARP inhibitor resistance. Interviewing Dr. Drew this evening, we have patient partner in research Linda Brown, and also back with us is Dr. Alicia Tone. Linda was diagnosed with ovarian cancer in 2018 and is an active member of the patient partners and research team. So Dr. Drew, I'll turn it over to you.
Great, thank you. I'll just try and share my screen. So if you let me know when you can see slides.
Yes, we can see it.
Yep, can you see that? And you can hear me okay?
Yes, all good.
Great. So I just want to thank you for inviting me tonight. And as has already been said, I'm going to talk to you about the NEOCATS trial, which is a neoadjuvant olaparib combination ovarian cancer targeted study. And before I sort of do that, I just want to introduce the topic with a couple of slides to try and sort of talk about why I want to do this trial and obviously the trial itself and some of the details. So I shared this recently at a previous presentation last week, and this is a quote from one of my patients in clinic who said to me, "Are you still using carboplatin and paclitaxel, Dr. Drew? My mum had that for her ovarian cancer 20 years ago." And obviously, my reaction was, well, yes, it works and nothing beats it. And we've always done it this way. But essentially, the backbone of, first-line treatment for women with stage 3 and 4 high-grade serous ovarian cancer is chemotherapy and remains chemotherapy. And many women are receiving chemotherapy in the first-line setting prior to surgery, so what we call neoadjuvant treatment. And that might be because the cancer is too advanced to be operated on. It might be because the patient is not fit enough at that point for surgery, or even due to delays in accessing surgery due to COVID and other sort of challenges following the pandemic. And the first line treatment, as we know, is carboplatin and paclitaxel, and some women get that with bevacizumab, and it's given every 3 weeks, and we usually give 6 cycles of chemotherapy. And if you're having surgery after chemotherapy, it's usually sandwiched somewhere in the middle. But how good is chemotherapy really? So these are this is the reality of the outcomes of chemotherapy for women that kind of get chemotherapy first. So the response rate from a recent trial, the ICON-8 trial, showed us that the response rate to chemotherapy was 61%. So response rate means what's the chance of the cancer shrinking on the scans. That doesn't include it staying the same. This is just patients whose cancers shrink with the treatment. When women have completed chemotherapy, on average, the progression-free survival, so the time for the cancer to return, is about 13.8 months, and that's after first-line treatment. And we know that 80% of women who get neoadjuvant chemotherapy, so chemotherapy first because their disease is too advanced, will recur and subsequently die of treatment. So this is showing that our first-line treatment is not that effective. Now, we have made great progress, and we've heard a lot about this tonight, looking at one targeted type of treatment, which is PARP inhibitors in selected patients. And what I'm talking about there is PARP inhibitors, which obviously we started out using them in the recurrent setting as upfront treatments themselves or as maintenance treatments, and then we've moved on to using them in the first line setting. And we've heard about SOLO-1 and the PRIMA trial, and most recently at ASCO just presented was the data for rucaparib, again showing a benefit in maintenance, and the benefit is the greatest in those women that have a BRCA mutation or this type of ovarian cancer that has homologous recombination deficiency, which includes those with BRCA mutations. And you can test that by biomarkers such as this thing called LOH-high or the Myriad MyChoice assay. And also platinum sensitivity is an indicator that you might have a homologous recombination deficient cancer. And this is the data from the SOLO-1 trial, and this is fantastic. But obviously, if you turn this on its head, you can see that the women at this point, we have 48% of women who've not recurred, which means that 52% of women have. So even with our best sort of first line treatment, we know that we need to do better. So are there any alternatives to chemotherapy that can be explored? And one of the things that I've been working on in a trial that Dr. Lee mentioned is the Mediola trial. And this is combining different targeted treatments without chemotherapy. And there is a lot of evidence in sort of laboratories and in sort of mouse models and in early clinical trials that these three drugs could work well together to enhance each other's activity on the cancer. And so I, with AstraZeneca, took part in this study called the Mediola trial, and there are lots of different parts of the trial, and this is just one of the parts of the trial. So in this trial, and this trial completed a number of years ago now, we took women who had not got a germline BRCA mutation, so they weren't women who had hereditary ovarian cancer, and they couldn't have had a PARP inhibitor or an immunotherapy treatment, and they had to have received less than 2 prior lines of chemotherapy, but they all had recurrent ovarian cancer, and they all could have gone on and had chemotherapy. But instead of doing that, they were recruited to the study. And we offered women 2 different treatments, and this isn't a randomized clinical trial, so these 2 cohorts of patients were recruited sort of sequentially. So women could have this triplet combination of olaparib, durvalumab, and bevacizumab, and I've just put on the right here what these drugs are, just to remind you. And then they could have had a doublet treatment of the olaparib and the durvalumab, so that's the treatment without the bevacizumab. And we were looking in this trial, it's a small study, and we were looking for what is the chance of this working, and is this are these triplet or doublet combinations safe? So I'm just going to show you the data for the triplet here, because that's what's being taken forward. So this is called a waterfall plot. I'll just go back one. And I don't know if you're familiar with looking at these, but each green bar here represents a patient's cancer responding to treatment. Anything that goes below 0 means that it's shrunk. Anything that goes above 0 means that it hasn't shrunk. And to be in response, you need to have a 30% or greater shrinkage. So if you look at this waterfall plot here, you can see that most women had a really good response to this chemotherapy sparing triplet treatment. So the response rate was 87.1%. And the length of that response was 11.1 months. But these are women with recurrent disease. So this is not a first line study. And we've submitted the long-term follow-up of these women to the ESMO conference, which will be held later this year. So I can't present that data to you today. So I said at the beginning that these were women that didn't have germline BRCA mutated ovarian cancer, but they might have had some other genetic abnormalities in their cancer. And we did this test called a genomic instability test on the cancer, which is similar to a HRD test. And we grouped the patient's response by whether they were GIS positive, so they had genomic instability, or they didn't. And what was interesting about the study is that regardless of the result of this test, women were responding. So this treatment seems to work across the board, whether you have homologous recombination deficiency or genomic instability or not. So that's interesting. And the other side of the study, and I'm not going to present all the safety data today, but the summary of the safety data is that this triplet was safe and there wasn't really any overlapping toxicity between these three drugs, and we didn't see any compounding of toxicity. So we saw what we would expect from these drugs as single agents, in the study, and most women were able to tolerate the treatment. So moving into the sort of neoadjuvant setting, which is the first line treatment, we've got a number of studies trying to look at sort of targeted PARP inhibitor-based treatments. And there are a few studies ongoing now in the US. One is looking at olaparib and the other is looking at talazoparib, and that's another PARP inhibitor, which isn't currently licensed or approved in ovarian cancer, but is approved in breast cancer. And these specific studies are looking at BRCA mutant or abnormal, ovarian cancer, or those with homologous recombination deficiency. So these are very specific studies, and they're very small, and we don't have the results of those yet. And this is the study that I have proposed and has recently been funded by Ovarian Cancer Canada and is supported by AstraZeneca. So this is the study of this triplet combination, and it's going to be investigated in women with advanced stage 4 high-grade serous ovarian cancer in a chemotherapy sparing approach. And there have been lots of patient partners, which I'm very grateful for, involved in the study. I've named the groups there. And also the study is supported with a big translational program and some collaborators I've listed on the top here, such as Connexia Health and Illumina Canada. And the aim of the study is to run it through the Princess Margaret Consortium through Toronto with Dr. Ami Oza's support, but hopefully to have the study open across Canada so that there could be the opportunity for many women to participate. Now, in terms of the study itself, what we're going to try to do is recruit women even before diagnosis. So if you have suspected stage 4 high-grade serous ovarian cancer, you may be able to consent to going into the study and having your diagnostic biopsy, but also the research biopsies as part of the trial. So it won't be just for women who've got confirmed disease, it will be for women with suspected disease. And that will be done as part of the sort of screening progress. And the reason to do this is that we're going to need additional biopsies from women, and we don't want people to have to go back and have a biopsy as part of diagnosis. If it could be done at the same time, that's being proposed. In terms of what's going to happen to all of these samples, so in the study, we're hoping to collect biopsies, we're hoping to collect fluid, ascites, pleural fluid, and then obviously women that go on to have surgery will be able to collect multiple samples through their surgical procedure. And these are just some of the things that we're going to do, but obviously I'm not going to go into detail with all of this tonight. The trial itself, this is what it's going to look like. So it would be for women with stage 4 disease who are planned for neoadjuvant chemotherapy and planned for interval debulking surgery. And we'd be asking these women to come into the study instead of having their chemotherapy. Now, this could be a risky thing to do because we know that we have a standard of care. So we're deviating from the standard of care. So it's important in the trial to be on the lookout early for those women that aren't responding. So after 2 cycles of treatment, women will have a scan. And if they're not sufficiently responding, and that will be decided by their own oncology team and on their scans, then they will come off this trial and go on to standard of care chemotherapy. Those women that are responding can continue with this triplet up until going for surgery. And then because this is a neoadjuvant study, everything that happens after surgery will revert back to standard of care. So the first thing that we're trying to show in this trial is can we not give chemotherapy in this setting, and is it safe, and how effective is it? So it's a small study that will involve up to 40 women. So there are some challenges ahead. So like all clinical trials, they are proposed by us as investigators, and we have to get funding for them. And we do have some challenges still with the funding for the trial. So bevacizumab is not a drug that AstraZeneca own, and they are not supporting that as part of the study. So they're going to provide olaparib and durvalumab for free, which is great, but we have to fund the cost for the bevacizumab. And there's obviously additional funds that we're still trying to raise for administrative support. So we have a lot of funding from Ovarian Cancer Canada, but there is still a shortfall in the funding for the trial. So I want to thank you all for listening. And as I say, I thank you to the patient partners, to Linda, who's going to grill me now on all of this. And I just thank you for having me this evening. And I'll stop sharing so that you can see everybody else.
Linda, you're on mute.
Yeah, sorry about that. So Dr. Drew, just before we start, I wanted to say that after watching some of your seminars and videos online and seeing your very extensive and significant list of research publications, how fortunate we are as Canadian women living with ovarian cancer to have had you move from the UK to Canada and bring with you your extensive and inspiring gynecologic research and expertise here. It's extraordinary and we're grateful. So thank you so much.
Thank you, Linda. I mean, I have to say you have probably Brexit and Boris Johnson to thank for that. But also the team here in Vancouver is a fantastic team, and I feel very privileged to be part of it now. So
yeah, so we're very fortunate to have you. So thank you. So I guess the first question would be, can you explain the current standard of care for women with newly diagnosed stage 4 or high-grade serous ovarian cancer, as opposed to stage 3? And why only women with stage 4 are at this point going to be in the NEOCATS study?
Yeah, so really good question. So the standard of care for women with stage 4 disease, it isn't particularly different to stage 3, although a lot of women with stage 4 disease have chemotherapy first and don't go and have surgery first, because obviously stage 4 means the cancer is more advanced and therefore may not be amenable to surgery. And the other thing that these women can access is they can access bevacizumab in combination with their chemotherapy if they have stage 4 disease, whereas those women with stage 3 at that point can't. So there are some subtle differences, but the kind of pathway is very similar. Now, the reason why we chose to focus just on stage 4 patients for this trial is that there's a number of reasons. Firstly, those women with stage 4 disease have the worst outcomes currently. So they have the greatest unmet need, in terms of improving treatment. But those women with stage 4 disease also have potentially disease that may be more accessible for biopsying. So they might have lymph node, metastases. They might have, other disease elsewhere that we can potentially biopsy as part of the study. And the other reason is that women with stage 3 disease in some centers might get surgery first, whereas in other centers they might not. And it's a bit more of a mixed group of patients. So we wanted to try to focus the study on a specific group so that we could make sure that there was kind of, homogeneity in the patients going into the study. So there's just a number of reasons. But in theory, where this study goes, if it's shown to be successful, it could be applicable to all women, with stage 3 or 4 disease.
That's terrific.
Hi, Dr. Drew. So you explain you call your study a proof of concept study, which I think you should explain what that means perhaps. And you're measuring safety, feasibility, and fitness for surgery. So perhaps you could elaborate on those things, especially how you're measuring monitoring safety and what you mean by fitness for surgery as well.
Yeah, so I mean, proof of concept usually is applied to a study where it's the first of its kind. So when we first developed PARP inhibitors, the first ever trial in a BRCA patient was called a proof of concept study, because it was like, do we even know if this is going to work? So although we have the evidence in women with recurrent disease that this is an effective treatment, that was a small study. So this isn't a 1,000-patient study. That the Mediola trial, these cohorts of women were very small. So we have to prove that this concept is applicable to women in first-line settings. So that's why it's called a proof of concept study. And obviously where there is a standard of care, it's very important that you protect patients from any harm. So what we want to do is focus on safety. And the first 10 patients that go through the study, their data and their results and all of the side effects will be analysed by a kind of independent committee, which will be formed to kind of monitor the progress of the study. And if there's any concern about safety, like there's too many side effects, or there's some concerns about people having surgery after this treatment, because that's what we don't know, how safe is surgery after these treatments, then we would want to stop the study early if there was a safety concern. Now, we know that giving bevacizumab before surgery is safe, but you have to stop the bevacizumab a number of weeks before surgery. So that will be happening as part of the trial. And we know that there's a study already giving a type of immunotherapy called pembrolizumab before surgery. And that study showed that it was safe. So, but what we don't know is the triplet, the 3 drugs together, is that going to be safe? So we have to make sure that we are kind of very early on making a decision about whether we recruit the rest of the patients to the study.
Linda, do you want to go ahead?
Yeah, and maybe we answered this question when we were talking about PARP inhibitors earlier. I'm not sure. So stop me, Dr. Drew, if I'm repeating myself. So your trial includes the use of a PARP inhibitor. at the very beginning of the treatment journey prior to chemotherapy and surgery. So are you concerned at all that that might lead to some kind of PARP resistance? Looking at the treatment journey down the road after surgery and chemo?
Yeah, so it's a good question. So it's unlikely that this study would do that because this study is just looking at 3 cycles of treatment before surgery. So it's a limited course. And then most of these women will be able to go on to their maintenance PARP inhibitor afterwards as part of standard of care. So I think it wouldn't be this study that would potentially cause resistance, but what we're actually trying to do is, which is the sort of opposite of that, is that we think, and we don't know this, that maybe the cancer is more sensitive to PARP inhibitors before chemotherapy. Now, we don't know this because it's not been shown in a trial yet, but there is some sort of scientific data to suggest that potentially it might work better if it's given first. But that's one of the things that the study's looking at.
That's exciting. Wow.
So Dr. Drew, obviously as one of the funders of this study, we're very excited about it and want it to be successful. So if it is successful, what would be the next step and what is kind of the ultimate goal that you're working towards?
Yeah, so I guess the evidence from the Mediola trial, shows that this triplet is very effective, and it's more effective than we would think of that chemotherapy would be as effective. But the Mediola trial didn't randomize anybody to chemotherapy, so it didn't compare in the same group of patients, how effective chemotherapy is. So it would be wrong of me to say this treatment is better than chemotherapy. So obviously what we're trying to do is introduce the idea of changing the paradigm and not giving chemotherapy with this small study. And if it is successful, then we would want to do a randomized controlled trial, comparing the triplet targeted treatment to chemotherapy. And that would need to be a much bigger study, and that would involve a lot more patients, and that would cost a lot more. So the first thing is to prove something small, and then hopefully expand it into a bigger study. So this trial in itself will not change standard of care, but it could be a step towards that.
So I think Steph is going to come on and has some questions, unless Linda, you have one you want to ask.
No,
Steph,
I just wanted to comment that it's such a huge paradigm shift. It's it's extraordinary and so very interesting.
Sorry, I was just going to close my blinds there. The sun is shining in here in Saskatchewan. We do have a couple of questions, Dr. Drew. So the first one here is, from what I recall, bevacizumab can affect bowel wall integrity. Would this not increase risk complications of bowel complications during or post-surgery?
Yep, so that's a good question. So we already give bevacizumab, as I say, as standard of care for this patient group. And we wouldn't give bevacizumab if we were concerned about the risk of what's called bowel perforation or fistula formation. So we already know who those kind of patients are, and those patients would have to be excluded from the study, but they would be excluded from bevacizumab as part of standard of care as well. So there's a specific things that we look at on the scans, to decide if somebody is at risk of complications of bevacizumab. But it is certainly something that will be part of the safety, of the study. So for example, if we saw more of that in this study than what we would expect with sort of standard of care, then that would be a red flag for the study. But we would be excluding women who we think are at risk of bevacizumab bowel side effects.
So on that, then another question just popped up here. So who would those patients at risk be?
So it's generally, so there isn't a sort of patient that you can define by the patient characteristics. It's more about the scan and where the cancer is. So if the cancer is very sort of matted onto the small bowel, or like, very much stuck on the bowel, there is a risk that you could get a perforation or a fistula. And these things just generally happen, or they don't happen. So it could be like never, it doesn't happen, or if it happens, it's a serious side effect. So it isn't something that the dose of bevacizumab, or you can't sort of protect patients from this if you give these drugs. So when we look at the scans, we normally discuss with radiologists and obviously surgeons whether we think this patient is at risk, and we would or wouldn't give them bevacizumab, and that happens routinely anyway. So anybody who was going into NEOCATS, if their own oncologist thought they were at risk of bevacizumab side effects, this side effect, then they wouldn't be going into the study.
Okay. And I just want to say thank you for so clearly answering all of the questions. I certainly appreciate it, and I'm sure the audience does as well. So thank you. And there's one final question. Are there any preliminary findings from the Athena trial?
Yep. So the Athena trial obviously was a trial a bit like the Prima trial. So it was looking at women who had stage 3 or 4 high-grade serous ovarian cancer, and they had their first-line chemotherapy, you know, after surgery, and then they obviously went on to a PARP inhibitor, which this time it was rucaparib, and again it was against placebo. And the data that's just been presented at ASCO is the data for the BRCA mutation, you know, carriers. So, and that shows, just like Solo 1 and just like Prima, that there is a benefit to having rucaparib. So it was the HRD and the BRCA patients. So it doubled the what's called the progression-free survival. So the time it took to the cancer to come back was doubled. So it's kind of showing us what we already know. And these trials, they're all designed at different times, you know, by different companies. And rucaparib was actually the first PARP inhibitor ever to be given to anybody in the world. But obviously it came in slightly later in terms of the ovarian cancer trials. So that's why we're seeing it reported now. But Athena also has some other arms, which is the immunotherapy treatment, and that data hasn't been reported yet. So we only got the rucaparib maintenance data at ASCO just last week.
Okay, excellent. Thank you. So that is
and I
all
think
of the
I would just say,
questions.
actually, Stephanie, sorry, I just think it's important.
Yeah, no worries.
And Anna said this before, and I think it's important to say it again. We don't consider as oncologists one is better than the other in terms of PARP inhibitors. You know, they all seem to perform well. They've never been compared directly with each other in a trial, and they probably never will be. So I think it's important, if you're on a PARP inhibitor, not to think, oh, there's a new one now and I want to go on that. That if you're getting a response to one, you're likely to get a response to the others. And if you don't respond to one, you're not likely to respond to the others. And it's just about what's approved and what's available. And I think that's the most important thing, rather than which one you're on.
Thank you for clarifying that. I'm sure a lot of people had that question, so thank you. And that brings us to a close. We have no more questions. So Dr. Drew, again, I just want to say that you're so easy to listen to and easy to understand. And I appreciate that. And I'm sure everyone else does tonight as well. Linda and Alicia, I want to thank you both too for sharing your obvious passion and your expertise and your experiences with us. So we truly appreciate that. And that brings us to the close of Ovarian Cancer Canada's Research Forum. We hope that you found the sessions helpful and informative, and that, like me, you can feel the passion and the dedication from these experts who are all working towards our shared vision to help people with and at risk of ovarian cancer live fuller, better, and longer lives. So the path ahead calls for added bench strength, and we're confident that our community of researchers, supporters, families, and friends will rise to the challenge. We've honestly come too far to stop right now or even slow down, as you've heard tonight. Thank you for your support and for further progress against this disease. Thank you for your support and confidence in this team. Without you, events like this and progress would not be possible. And with that said, we want to know what you thought about tonight's event. So we would love it if you could take a few minutes of your time to fill in the evaluation form. We are going to link that in the chat right now, and you also will receive a link via email. Your responses really do help us shape future sessions and ensure that we're giving you the programming that's relevant to your needs. Also, I just wanted to remind everyone that we have recorded all of the sessions tonight, and our goal is to have those up on our website by the end of next week. If you have any questions about any of the sessions tonight, please also feel free to reach out to us, and we can have our information put in the chat as well. And if you want to learn more about Ovarian Cancer Canada's research initiatives, you can find that on our website at ovariancanada.org/research. And if what you heard today inspires you to further progress, please consider making a donation. Progress is only possible with your support. So again, I want to thank all of the fabulous speakers we had tonight. A huge big thank you to the patient partners in research and the Teal Sisters. What a powerful way to get information across and ask questions of our expert, for sharing your wisdom and your lived experiences with us. And thank you also to everyone who attended tonight, our community, for joining us. I want to wish you all the best. Take care, and good night from all of us here at Ovarian Cancer Canada.
Yeah. Good night.